I'm Henrik Dohlman. I'm the chair of the pharmacology department, and Yinglong asked me to welcome the participants of this meeting. We were very happy to support it logistically and financially. I think this type of work is going to be increasingly important in the biological sciences generally, and in pharmacology in particular. And I really want to thank Dr. Miao for organizing this workshop. It's a great service to the community. And so let's give him a round of applause for his organization. So I hope you enjoy your day. I hope you learn a lot. I hope you enjoy, those of you who are here in person, I hope you enjoy your time in Chapel Hill. And thank you for participating. Thanks.
All right, good morning, actually good afternoon, good evening. So it's better for those on Zoom. So next, I would give you a brief introduction about our workshop and the GaMD. Before I talk about the GaMD method, so let me try to move this. So can actually everybody hear me okay on Zoom? Okay, sounds great. So before I talk about the GaMD, I want to thank the people who make this possible. First, I want to thank our 5 invited speakers, Professor Shirley from UC Davis, Professor Ramon Quintana from University of Florida, Professor Steffen Lindert from Ohio State, Professor James Prestegard from University of Georgia, and also Professor Pengfei from Loyola University Chicago. And I want to thank our wonderful team members in the lab, especially Jing'an, Kia, and Victor. So they would give hands-on tutorials in the afternoon. Amy also went through all the tutorial documents. So that's lots of work. Yeah, thank you. Then, yeah, we really appreciate the support from our pharmacology department and also computational medicine program. Yeah, Henrik already gave the opening remarks. Professor Tim Elston, he's a co-director for CommAD program, and also our wonderful staff, Q, Victoria, and Mimi. And I want to thank our UNC Research Computing Director John McGee, Rob Zeld, and CD. Without them, this workshop will not be possible, so I'll not be here today. So thank you so much, and let's give them a big applause. Now, I want to thank all of you for being here. For the registration, we received about 140 responses. We have about 55% graduate students, 22% postdocs, 8% undergraduates. I also have senior staff and principal scientists, and also faculty. We have people from both academia and industry. For the experiences, about 67% heard about GaMD. A quarter learned about GaMD, and about 30% used the GaMD in research. And for the interest, like more than 80% want to learn about the ligand GaMD or liGaMD. 66% for peptide GaMD, and 77% for protein-protein interaction GaMD. Well, thanks so much for your interest. I would say, like, we'll provide tutorials for these methods in the afternoon. I know like many of you have different time zones, so it can be later for some of you. So we really appreciate you being here. Thank you. Next, I would give a very brief introduction about the GaMD. GaMD stands for Gaussian Accelerated Molecular Dynamics. So it's an enhanced sampling technique. It adds a harmonic boost potential to reduce our system energy barriers. As we know, our proteins have different low energy states. Let me try to find my laser. Like a bound, unbound, folded, unfolded. In between, you have these high energy barriers. Now, given reference energy E, when the cis potential drops below that, we add a harmonic boost potential. So this is our formula. With this boost, we can raise the low energy wells, reduce the energy barriers, then we can speed up repeating summations. In GaMD, a boost potential has a narrow range. Choose a Gaussian distribution. Then we can apply cumulative expansion to the second order, or just a Gaussian approximation to rate our summation. I want to calculate the original free energy. It's the modified minus the boost potential accumulants. For the accumulants, the first order would be the average. Second order would be the variance. With this, we can calculate the accurate free energy profiles from the GaMD summations. Well, in comparison, conventional is like hiking. So you walk step by step to climb mountains. It's fun, but can be very slow. Every step you walk could be 1 over a few femtoseconds. That's 10 to the minus 15th of a second. Even as you walk like a billions of steps, you reach only microseconds. In GaMD, like we fly on a helicopter, we lift ourselves. We have like a much higher potential. Then you can fly over many mountains very quickly. And then you can enjoy like many more views. So by running short microsecond summations, we can capture long timescale events, like over milliseconds. So I've shown this in a number of previous studies, especially for GP cells and CRISPR-Cas9. All right, can everybody on Zoom can see the slides? Okay, sounds good, yeah. All right. Sorry about that. So yeah, in GaMD, yeah, we can fly on helicopter. It's much more efficient. So we can run like short summations to capture long timescale events. Then we call GaMD a time accelerated computational microscope. Since 2015, it has received more than 1,000 citations by people around the world. So I want to thank many of you in the audience. And then we have GaMD coded in a number of popular packages, including Ember, NumD, OpenMM, Genesis, and Tinker. GaMD has been used to study like protein folding, conformational change, ligand binding, like in the movies here, interactions between proteins and other biomolecules, like peptides, proteins, carbohydrates, and nucleic acids. It's also applied to study protein n-ary and explore different protein structures for drug design. This is what we learned like 2 weeks ago. So GaMD has been coded in Discovery Studio. They also offer a webinar, like how to run faster molecular summations. I think the link is still active, and you can check it out. So let me talk about the GaMD workflow in the Discovery Studio. Based on GaMD, we have also developed new selective GaMD algorithms. These methods are much more efficient for us to study the dissociation and binding of like small molecules, peptides, and proteins. Our first method here is called ligand GaMD or liGaMD. The basic idea is to add a selective boost on the ligand. So this is for the non-bounded interaction energy between the ligand, the protein, and also the environment. With this boost, we can push out the ligand and speed up the ligand dissociation. In addition, we add another boost to the rest of the system. This includes ligands and the solvent. Then we can speed up the ligand diffusion. That can help the ligand rebinding. With the method, we test this on protein ligand binding. Our model system is trypsin. In the crystal structure, this protein binds inhibitor, benzamidine. Benzamidine has a positive charge. It can bind aspartate at the protein active site. In our system, we also added a number of ligands in the solvent. But here, I would hide the solvent so that you can look at the protein and the ligands more clearly. As during the submission, the ligand could dissociate and rebind. As you watch the movie, this is like a soccer game. Our ligand would be the soccer ball, right? Now I have one difference. Instead of two teams, you have just one team in the field. All the players in the field can work together to score the goal. Well, once that happens, you need a goalkeeper who can kick the ball out of the goal. Otherwise, like the ball would be stuck in the net. Well, in this regard, like, LiGAMD is very efficient. It's for the first time we can say both the ligand dissociation and rebinding in just 1,000 nanoseconds, 1 microsecond submission for this model system. Then we had 5 similar 1 microsecond submissions. In our submissions, we recorded the boost potential for every submission frame. Then we have algorithms to take out the boost. We can recover the original ligand binding for energy and kinetics. So this includes delta G, K on, and K off. Compared with the experimental data, our estimates from the submissions are pretty accurate. At the same time, we can also calculate the kinetic rates directly from the submissions. We call this accelerated rates. If you look at the numbers, we can greatly speed up the ligand binding and the dissociation. Especially for the dissociation, it's by about 7 orders of magnitude. So that's about 10 million times. Well, in experiments, the half-life of this ligand is about 1.7 milliseconds. In our summation with the acceleration, we can reduce that to just 29 nanoseconds. So that's why we can score multiple goals in just 1 microsecond. If you run conventional MD, you may never score a goal because our same time, like microseconds, are much shorter than what have in experiments, milliseconds. Apart from ligand binding, we're also interested in peptide bind and protein-protein interactions, but they have very unique features. For the peptides, they're extremely flexible. They can essentially fold during the binding process. For the protein-protein interactions, the proteins would be much larger. They have well-defined structures to bind and form complexes. And then we like to add different boost potentials to these problems. For the peptides, we add a boost on both the non-bounded and also bounded potential of the peptide. In this way, we can model the peptide flexibility and also speed up the peptide dissociation. For protein-protein interactions, we would add a boost on the non-bounded interaction energy between the two proteins. So this includes the electrostatics and the van der Waals interactions. With this boost, we can also speed up the protein dissociation. Similarly, GaMD, in both methods, we add another boost to the rest of the system, so they can help with the rebinding of the peptide and the proteins. For the method here, for the time here, I would focus on like the PPI-GaMD. So our model system would be the binding of bar star to the barnase. With the crystal structure, I will perform the 6 independent 2,000 nanosecond submissions. From our submissions, we looked at the RMSD, or root mean square division of the residues we found at the protein interface. Compared with the crystal structure, say that two proteins can bind and dissociate multiple times in each submission. Then we can also calculate the protein binding for energy and kinetics. Again, compared with the experimental data, our estimates from the submissions are pretty accurate. And as you look at the accelerated rates, we can greatly speed up the protein-protein interactions. especially for this dissociation, it's by about 13 orders of magnitude. So that's about 10 trillion times. I think that's really remarkable. I mean, in experiments, the half-life of this protein complex is about 1.5 days. But in our simulation, we can reduce that to just sub-microseconds. So I would say like PPI-GaMD is very efficient. And I think Kia in the afternoon will give a tutorial on the PPI-GaMD. Recently, also came up improved the liGaMD algorithms. So these are spearheaded by Jing'an in the lab. As we apply the liGaMD on more problems, we noticed its limitation. So this is for tiflizine. It's another model system of protein ligand binding. If you look at the ligand, benzene, it binds deeply inside the protein. So the protein pocket would be closed on the surface. And then with the liGaMD, so no matter like how much acceleration we add to the ligand, it's like a confined and it cannot dissociate. And then we came up with this improved the approach. We add a selective boost on both the ligand and also the surrounding residues in the protein pocket. So these are highlighted in blue here. Then we can sample the protein flexibility and speed up the open the protein pocket essentially. And then the ligand could dissociate and rebind like in the movie here. In just this year, we also published the liGaMD 3. It works for both small molecules and flexible peptides. Instead of only 2 boost potentials in the previous methods, we add 3 boost potentials here. We call this triple boosts. So one is for dissociation of the small molecule peptide, another for rebinding, and then the final one for system conformational changes. Our model system would be the MDM2 protein. So this is a critical target for treating human cancers. It can bind both the small molecules and the peptides. Then with the liGaMD 3, I would say we performed 3 independent 2,000 nanosecond simulations. As you can see, in each simulation, we observed many times of dissociation and binding of both the small molecule and also the peptide. On comparison, we also performed the simulations with the liGaMD in the same length, but we observed much fewer dissociation and binding events for the ligand and peptides. So in this regard, the liGaMD 3 is much more efficient. And Jing'an in the afternoon would give a tutorial about the liGaMD 3. So that would cover both small molecules and peptides. On a brief summary, we have developed a selective GaMD. This includes liGaMD, PepGaMD, and PPI-GaMD. By adding selective boost potentials, we can speed up our summations by millions to trillions of times. Then by running only microsecond summations, we can capture both the dissociation and the binding of both the small molecules, peptides, and proteins. Then we can calculate the binding free energies and the kinetics. These include delta G, K on, and K off. As you know, these are really critical parameters for therapeutic design of drugs, peptides, and antibodies. So that's my brief introduction of GaMD. And then this slide shows a schedule for workshop today. So I hope you will enjoy the day. And for the rest of the morning, we have 5 invited talks. And for the moment, I think we have time. Do you have any quick questions about a GaMD?
Hello, MDANCE, que você tinha falado aquele dia. Vai falar às 9h50.
Can you speak louder, please? Or do I have a way? Any questions from the Zoom? Yes, please.
Thank you for the talk. I'm wondering if by modifying, if you introduce this artificial boost in the pocket with the ligand, if somehow if you, I wonder how much you can trust the mechanism of the dissociation.
So yeah,
Like how much can you trust it? How can you trust that this is actually how the ligand dissociates?
that's a
Pocket.
Yeah, that's a wonderful question. I would say by tracking ligand binding, or especially for the rebinding, if you know like the ligand can go back to the target site, just like in a crystal structure, so that'd be reliable. On the other hand, in terms of the theory, yeah, I don't go into all the details, but we can we add the boost potential, but you still need to have the original shape of the energy surface. We don't add too much, so the perturbation would be very small. So the path would be still reliable. We test this on a couple of model systems, for like tifloizine, trypsin, and even for like GPCRs. So the path we got from binding, they are pretty similar to those observed in Anton, like conventional MD submissions. Yeah. So would that answer your question? Okay, great.
So you've got times listed on the bottom of these, and they're accelerated times.
Yes.
Sorry, how do you get the estimate of those times? And going from, a conventional MD that's a microsecond to accelerated that, is a millisecond.
So as we get to selective GaMD, like a ligand GaMD, we can recover the kinetics. So we can really calculate the K on and the K off. So we have like a theories to recover those rates. They can really relate the time. Yes. Yeah. Okay. Well, thank you, and I think it's time to move on. Our first invited speaker would be Professor Steffen Lindert from Ohio State. Go ahead. So Steffen obtained a PhD in chemical and physical biology at Vanderbilt. He had a postdoc with Andy McCammon at UCSD. Steffen started his lab at Ohio State in 2015. His lab develops new computational techniques for modeling biological systems, and also apply these methods to predict the protein structures and discover new drug molecules. I think Steffen will move to UCLA in just July next year, so that'll be very exciting. And for today, yeah, he will talk about the calcium modulation of cardiac troponin C by small molecules. With that, yeah, I will turn it over to Steffen. Give us 10 seconds.
Thank you so much for the kind introduction, Yinglong, and also thank you so much for inviting me to give this lecture. I hope on Zoom you're seeing the full screen. So I will talk today how we're using both free energy methods and then MD and Gaussian accelerated MD, and I will point that out wherever relevant to actually look at contraction in cardiac muscles. So the system of interest that I will talk about today is cardiomyocytes or heart muscle cells. These are incredibly complex systems with like lots of ion channels on the cell membranes and this sarcoplasmic reticulum, this calcium storage, intracellular calcium storage. But I will focus on a protein complex involved in this contractile machinery, namely troponin. If we're zooming in on the sarcomere, the contractile machinery here, what we can see is that it's actually an alternating collection of thin and thick filaments, where the thick filament is myosin and the thin filament is mostly actin, but also some tropomyosin and troponin C and T and I. Try to mute. Okay, I'll try to power through here. Calcium binding to troponin is actually initiating contraction in that it allows for, I think I have this on the next slide, it allows for this tropomyosin molecule, which in the resting state is actually blocking cross-bridge binding site. It's upon calcium binding to troponin, a bunch of molecular events happen that allow for the tropomyosin to slide away, expose these cross-bridge binding sites for myosin heads to bind, perform a power stroke, and thus contraction to occur. We know a lot structurally about troponin. There's a 3 subunit complex where we have the troponin C subunit, the calcium binding subunit shown in blue here, then the inhibitory subunit, troponin I, shown in reddish here, and then also more structurally important troponin T subunit. I will focus almost exclusively on troponin C, and there's a bunch of molecular events that we will look at today, and that is calcium binding to the calcium binding site 2 in troponin, then the opening of a hydrophobic patch between 2 helices in troponin C, A and B, and then the subsequent binding of the switch peptide from the troponin I subunit. So we'll focus exclusively on this. And I just want to quickly spend a minute on talking about this opening of this hydrophobic patch between these 2 helices A and B that I mentioned in troponin C, which can then facilitate the binding of the switch peptide. The way that we measure this opening of the hydrophobic patch is an interhelical angle. And it turns out that the way this is measured, the numbers are counterintuitive in that actually the higher the number of the angle, the more close the hydrophobic patch. So structurally, we know that in cardiac troponin C, the closed configuration is at an angle of around 100, interhelical angle of around 135 degrees, while an open configuration corresponds to 90 degrees. Okay, so our goal for the studies here, and seems somebody has drawn in the slide here. But yeah, so our goals for these simulations is to use MD simulations and free energy methods to quantify calcium binding, hydrophobic patch opening, and also switch peptide binding. And once we have sort of confirmed all of this for like wild type systems, we want to then study the impact of mutations and small molecules. And our long-term goal here is to identify mutant or small molecule calcium sensitizers that could be used in the treatment of heart failure. And I'll talk more about the small molecule drug discovery later on in the talk. So this is a schematic of the proposed energy landscape of these crucial molecular events in troponin C, where you actually have that first step, which is calcium binding to the apotroponin C, which is an energetically favorable process, right? Then the opening of this hydrophobic patch between these helices A and B, which is an energetically unfavorable process, right? So you don't want these hydrophobic helices exposed to solvent, which is then stabilized by actually binding the switch peptide from troponin I. And the goal here is to actually use these free energy methods to map out these different stages of the free energy landscape. And I'll start first by looking at this opening of the hydrophobic patch, for which we decided to actually use umbrella sampling simulations. The way we did this is we used steered molecular dynamics to get different frames in all states of openness. I'm showing here sort of the different frames every 2.5 degrees that we got from steered molecular dynamics all the way from open to closed configuration. We used, in these simulations, we actually used the interhelical distance, which is the distance between the beginning of the A helix and the end of the B helix, as a proxy for the interhelical angle, because you know, the way we did this is in, thank you so much, in MD, you have to use certain reaction coordinates. Interhelical angle isn't one of them. And so we used interhelical distance. We showed that there's actually a very strong correlation between interhelical angle and interhelical distance. So this is a good proxy to use. And then we picked frames going from 140 degrees to 90 degrees in 2.5 degree fragments. We then ran umbrella sampling simulations, and this is just to show that the individual windows that we used for the umbrella sampling simulations are clearly overlapping, both in terms of like the interhelical angle as well as the interhelical distance. And we were then able to use a weighted histogram analysis method to deduce the free energies of opening of this hydrophobic patch for calcium-free troponin C as well as calcium bound troponin C. I'm also showing this for the skeletal isoform. Let's just ignore this for today. I don't have enough time to talk about that. So and what we saw is that we got about a 14 kcal per mole energy difference in the calcium bound system, because calcium binding is a crucial first step in actually being able to open this hydrophobic patch. So we were able to assign about 14 kcal per mole to this particular part of the free energy. We could also then repeat the simulation in the presence of the switch peptide, in which, not surprisingly, right, you're actually seeing that now the open configuration is a more energetically favorable. Doesn't make sense, right? Because it's hard to close this if the switch peptide is in there. And we got a free energy of about -7 kcals per mole. So that assigns these 7 kcal per mole here. So once we had done this for the wild type, we were actually able to look at different mutations. So we chose a number of mutations that have been associated with cardiomyopathies, namely dilated cardiomyopathy or hypertrophic cardiomyopathy, DCM or HCM, related mutations in the N-terminal region that we're interested in. So we chose to study these disease-related mutations. We, in addition to that, also investigated one designed calcium sensitizing mutation, L48Q. And the question is, well, what effect do these mutations have on the energy landscape? We first looked at the HCM mutations. And here's again showing our free energy of opening for the wild type system. And what we could see is that all mutations that are either associated with HCM or the calcium sensitizing mutation, L48Q, all lowered this free energy of opening. This is absolutely consistent with what is known about HCM, or of course the calcium sensitization of L48Q, in that it makes it easier, right, less energetically costly, to open the hydrophobic patch, consistent with the calcium sensitization effect of these mutations. On the other hand, if we looked at these DCM mutation, we saw exactly the opposite effect, right? They actually increased the free energy of opening in all 3 cases that we investigated. That doesn't mean that they're not also affecting the calcium binding affinity or the switch peptide binding affinity, but they're certainly having their calcium sensitizing or desensitizing effect in part by making the hydrophobic patch opening more energetically favorable or disfavorable. We subsequently also looked at the effect of small molecules on the opening of the hydrophobic patch. There are a few small molecules known to bind the hydrophobic patch. These have been designed as part of drug discovery efforts to find small molecule calcium sensitizers. They've also focused on binding in that hydrophobic patch. I'm showing you a bunch of structures, either NMR crystal structures of, say, Baprodil, for instance, or W7, experimental structures, and or docked poses of some of the molecules that we don't have experimental structures for yet. These molecules have been designed to be calcium sensitizers. However, most of them actually, and I'll just say this in advance for my second stage of the talk later today, is they don't really have very high affinities, right? So most of these actually bind in the hundreds of micromolar. So very, very low affinity molecules. So there's a lot of room for improvement. But we wanted to just see, well, given that we know that they're actually binding troponin C, how do they influence the opening of the hydrophobic patch? So we redid all these steered MD simulations as well as umbrella sampling in the presence of the small molecule. And what we saw is that, here again, just for comparison, the wild type value without any small molecule, the one with the switch peptide bound, and now again, just calcium and small molecule bound systems, is that we saw a dramatic lowering of the free energy of opening. So whatever energy barrier we have here in the wild type system is lowered dramatically if you have a small molecule in there. And that is consistent with them kind of binding to the hydrophobic patch, keeping the hydrophobic patch open in a semi-open configuration. So once calcium binds, it makes it much easier to open up completely and still bind the switch peptide. So that was really great to see that we could do this for the hydrophobic patch opening, both for mutations as well as small molecules, and since using this for protein design studies, for which I don't have time to talk about today. But I will quickly also talk a little bit about how we actually explored the free energy landscape of these two other molecular events. So we looked at calcium binding free energy. This is, we decided to use thermodynamic integration for this. So this is a alchemical method where you're essentially disappearing the ligand, calcium in our case, by removing electrostatic and van der Waals interactions in its binding site and then reappearing it in a box of water to complete the thermodynamic cycle, and that allows you to calculate the free energy of binding. We got values for wild type as well as one mutant system. This is work in collaboration with the Tibbets group at Simon Fraser University, in perfect agreement with experimental values at about minus, for the wild type system, at about minus 7 kcals per mole. We also attempted to do the same with magnesium binding for these two systems. We got the right trends, but the magnesium parameters aren't good enough to actually get realistic values. We finally also looked at the binding of the switch peptide and used actually umbrella sampling simulations again, as this was just isolated switch peptide and the N-terminal region of troponin C. And in agreement with experimental values, we got binding free energies of about minus 5.4 kcals per mole. And then again, looked at different mutants, including this calcium sensitizing mutant L48Q again, which according to our simulations, in addition to making it easier to open a hydrophobic patch, it is also having an effect that it has a stronger binding affinity for that switch peptide. This again, we're using in protein design studies as we speak in collaboration with the Reiser and Davis groups at OSU. Finally, I want to say that while everything that I talked about today was exclusively focused on the N-terminal domain, we've also done simulations on parts of the whole thin filament, where we actually had actin, tropomyosin, and the whole troponin complex there, and we looked at the binding of the switch peptide in this environment. This is the unbound state in which we then used umbrella sampling to completely flip over the switch peptide and bind. And we saw binding affinities of about minus 8.9 kcals per mole. Again, in agreement with what has been shown experimentally, that there is a higher affinity in the complex of the whole thin filament. So this was very encouraging. Again, we're using all of this for protein design studies. I will finish today by talking a little bit about our drug discovery studies and our use of GaMD within there. And we're doing drug discovery for heart failure. This is whenever sort of your heart doesn't pump blood strong enough to meet your body's needs. Millions of Americans suffer from that. There is many different ways to address this. One way that has been argued would actually be to increase troponin sensitivity to calcium as a possible treatment for that, right? And the way that we approach this is we actually, at the start of this, we looked at all known calcium binders to troponin C. This is a list of the ones we had at the start of the project, so 10 different molecules that we knew bound troponin to different degrees of affinity, but we knew they bound. And the idea was, can we use knowledge about these molecules to actually identify receptor conformations that bind most strongly? And this, just to repeat, we're focusing again on finding molecules that can bind in this hydrophobic patch region that I talked about long before. In order to account for receptor flexibility and get as many different conformations, what we did is we collected 206 possible configurations, many of them from crystal structures as well as NMR, but also from Gaussian accelerated molecular dynamics simulations. And I just have a quick slide on the details of these GaMD simulations. We actually 4 representative conformers of these 3 different proteins. We ran 300 nanoseconds of Gaussian accelerated molecular dynamics in amber. These 3 proteins, or like PDBs, were chosen because they contained the largest ligands, Baprodil and W7, and then 1MXL actually was on the other end because it was a structure that didn't have a ligand at all, to try to get a range from large pockets to smaller pockets. We subsequently clustered these simulations to obtain 10 representative receptor conformations from each of them, giving us a total of 30 extra receptor conformers that we derived from GaMD. We then had our ligand test set. These were those 10 compounds that we know to bind troponin C. And then we selected 1,000 decoy compounds, presumed inactive compounds. We went to great lengths to make sure that those, in terms of molecular weight, volume, and many other properties, actually kind of had a very similar distribution. These decoys had a very similar distribution in molecular properties to our actives. And then what we did is we took all 1,010 compounds, so 10 actives, 1,000 decoys, and docked them into each of those 206 troponin conformations and ranked the ligands by docking score. We used GlideSP to do that. And I'm showing you here, this is a cartoon representation, not actual data. Sort of once we had this ranked, right, really what you would like to have is something that looks like this, right? That has as many of the active compounds, the ones that you know actually bind troponin C, at the top of the list versus those decoys. They should be further down in the list. We used receptor operating characteristics curves for evaluating this more quantitatively. So this is a way to plot where you're pretty much going a step up in the curve. You're starting here, ending at the top end. Now my mouse is gone. And here, and you're going a step up anytime you find an active in your list, and a step to the side anytime you find a decoy in your list. So in the most optimal scenario, in case you ranked all your 10 decoys at the top, you would go right up here and then over. So the area under that curve of this rock curve is actually an assessment of how much do the actives rank ahead of the decoys. The random distribution would be that dashed line here. What you can see is that some of the receptor configurations that we saw actually had area under the curves closer to 0.8, which is quite a very predictive. We also looked at enrichment factor, which is just the number of actives within a certain fixed number of very top scoring compounds that we can test experimentally. We chose 40 there. And in this particular case, we actually find 6 of the 10 actives within the top 40, which is also quite phenomenal. So we identified the 3 most predictive receptor structures. In this particular case, I have to say, and I will talk about in a couple seconds, that we have other examples where that's not the case. In this particular case, unfortunately, none of the GaMD structures was among them. We then subsequently docked a small molecule library, hundreds of thousands of compounds into these three most predictive conformations. We identified the hits. Our experimental collaborators in the Davis and Reiser groups tested those. We then did ligand optimization to finally find something that we refer to as compound 5, which was quite great. It actually had an increase of the, quite dramatically increased the off rate of calcium and had an apparent binding affinity of just 1.5 micromolar, placing it really among the most potent compounds that we've ever found. I should also say we've since actually improved this, and we're now in the nanomolar binding affinity, which is again orders of magnitude better than anything that we've seen to troponin C so far. And I just want to also say that we have tested this compound since in actually red cardiac trabecula, as well as also slow skeletal muscle fibers. And what we saw is that we see a notable shift consistent with calcium sensitization, even in these muscle fibers of compound 5, and that also has an effect in slow skeletal muscle. So we're really going all in on improving this compound. I just want to end. So this is the hit improvement that I mentioned. Is we do this in collaboration with also Dr. Karakash Bristow at the West Virginia University, and we're now solidly in the nanomolar binding affinity. Just want to end really quickly that we've actually shown that we can generalize this principle of actually identifying predictive receptor conformations by looking at 20 different target systems. And what we did is for each of those 20 target systems, we looked at different conformations from the protein data bank as well as from Gaussian accelerated molecular dynamics. We took all known binders. We split up these binders in different sets, active set A, active set B, and docked them in individually. We then looked at what's the area under the curve for active set A, active set B. And what we saw is generally a very, very good correlation between these two. What that means is that actually knowledge of just a few active compounds really makes it possible to find receptor conformations that would then accurately predict other known compounds. And here, this is just, again, where I'm plotting the actives, the ROC curve, the area under the curve for the ROC curve of active set A against active set B, right? So this is quite impressive that this shows us that if you find something that is predictive with just a few of the actives, you can pretty much get the other actives for free. And I just wanted to say is in that case, we also clustered 25 separate 300 nanosecond GaMD simulations. And actually for these three kinases here, we successfully generated conformals that ranked among the top 3 most predictive configurations, which really underscores how important GaMD is for identifying conformations of proteins that can be used in drug discovery that are likely going to be more predictive to actually finding actual binders than anything that you can find in the protein data bank. So that is something that I want to stress is certainly something you should be doing if you're looking at any kind of receptor flexibility. Do throw in GaMD simulations. They're incredibly helpful. And I just want to mention, we also, for a subset of the targets that we looked at, actually 5 of them, we picked ones that were all cancer related. We tested the top 50 compounds that we got from docking into the most predictive configurations. And we actually identified 22 novel inhibitors, all in the low micromolar to low nanomolar range, with our most potent inhibitor actually being a single digit nanomolar inhibitor. So again, that just underscores that this receptor identification is incredibly helpful, and Gaussian accelerated molecular dynamics is a really important tool in that. And with that, I'd just like to thank everybody in my group that did this, everybody that gave us money. And as Yinglong mentioned, we're moving to UCLA. So if you're on the call and you're interested in computational biochemistry, we're looking for graduate students and or postdocs, and I'm happy to take any questions if we have time.
Struck by the size of the free energy differences between your open and closed conformation, which some of the fields are really high, 20 kilocalories per mole. And if the ligands find some of these are tiny binding energy. So I guess maybe the answer is that the important thing is actually the kinetics of going on and on. That changes dramatically, but the actual percentage of states that minuscule.
That's a good point. I should also say that while I'm relatively sure that the binding energy of the switch peptide is actually accurate at about 7, we do not have experimental measurements for the hydrophobic patch. So I totally give you, this might also be an overestimation. However, what we have seen is that at least with looking at like these DCM and HCM mutants, we do think that at least we get the trend correct, right? That whatever we measure, even if it is an overestimation, we would probably, if we are sensitizing it, we would be able to see this in our free energy simulations. And that, at least for our protein design, that's the most important part.
I have a question on Zoom.
Question on Zoom. What's the question?
How were the decoys obtained?
How were the decoys obtained? We actually, there's like many different decoy sets. We tried several of them, and I think we did end up going with a subset of the Schrödinger decoys that are available. One of the sets, yes.
Before you talk, and related to the same question, what were the AUC values for CT and C2 and 3, where you had more decoys than comparing to the active compound and the top rank compound?
It says again, which value are you, the troponin stuff or the?
The one where you has shown the decoy and active ligands.
For troponin?
Yeah.
Okay.
The other, I don't remember the slide number, but it's the same slide. Where, yeah, this one.
This one, okay.
But this one for the 2 and 3, what were the AUC values or?
For the. So wait, so sorry, this is just cartoonish, right? This is, I don't think this is actual data. I just wanted to make an example where there's a lot of actives at the top. But what would be the case is to just draw you how this would look. This is actual data, right? This would look if you had all 1010. Within the first 40, you would have 6 green ones. And because we do see this going in a little bit, you would probably have the first 3 or 4 to be red. And then it seems there's a bunch of, I mean, kind of 4 of them, right, coming up relatively quickly. So maybe within the top 10, you have 6 red ones and 4 green ones.
Maybe one more quick question.
Yeah, really great work. I was curious about the free energy calculations, specifically the TI calculations on your calcium binding. We're just using like the gap force field.
Yeah.
I guess did the force fields, like different force fields, come into any consideration with treating calcium, especially because your magnesium results weren't as
exactly. So yeah, we should certainly, if we wanted to do this in a more general way, we should use different force fields. Maybe even look at like polarizability or things like that.
Yeah, I was just curious about that because
we haven't tried it. Yeah, because also in a sense, right, the point that where we really want to use this force then assess small molecules and or like designed versions. And as I was saying before, the absolute values aren't that relevant for us as long as we get the relative free energies accurate, you know. Yeah.
But because of the time, yeah, we may have to stop here. But let's thank Steffen for the wonderful talk.
If you, I'll log into Zoom and hunt down that question.
All right, thank you.
Thank you.
All right, our next speaker would be Professor Ramon Quintana from University of Florida. So Ramon obtained a PhD in chemistry from University of Havana. They had a postdoc at the McMaster University and then York University with the Polanyi Prize. He started his lab in chemistry at the UF in 2020. His lab develops new ab initio electronic structure methods They study charge and spin transfer in chemical reactions and solvation processes. They also work on clustering algorithms based on extended similarity. Ramon has received a number of awards, including the Oak Ridge Ralph Poey Junior Faculty Enhancement Award, the OpenMM Outstanding Junior Faculty Award, and also a MIRA R35 Award from NIH. Today, he will talk to us about MDANCE. So it's a flexible and versatile clustering package. So without further ado, I will turn it over to Ramon.
So thanks, Yinglong, once again for the invitation. Thanks, everyone, for being here today. Thanks for the organization. So I'm going to be talking about essentially what to do after you are done with your MD simulation. So you usually have this like huge amount of data that you generate after running one or multiple simulations. And then what are some of the protocols that you can use to navigate through that in a way that you don't spend as much time analyzing your data as you spend essentially running your simulations. So my group has been putting together a package essentially led by Lexing, one amazing grad student in the group, that is called MDANCE. I guarantee you it's not going to be it's one of many amazing acronyms coming your way in the talk. So with MDANCE, we're essentially putting together a package to do post-processing analysis for MD simulations. At a very broad level, we have essentially ways for you to interface with your favorite codes, let's say Amber, GROMACS, NAMD. So you pick your tool to run MD. We can interface MDANCE with that. We have many different ways to process your methods, to process your results, especially something that we're really focusing on and was mentioned by the previous speaker is how to do clustering. How to do clustering in better ways, in more robust ways, and in faster ways. I am mostly going to be focusing on methods for biomolecules. Nonetheless, at the end, I am going to include two very short slides. If you are doing a small molecule analysis, we can also help you. If you're doing drug design, these tools can also be really helpful for you. So the key idea behind MDANCE is that we are using a new way to quantify distance or like separation between molecules, between conformations. Usually, and this is something that was like that this appears in Yinglong's presentation, appears in the previous speaker's presentation. Usually, you are used to using RMSD to quantify how separated are two different conformations. However, while this is perfectly fine once you have only two conformations, if you have many conformations, taking the average of all of those RMSD values is essentially going to be an O(n)² problem, both in memory and in time. And if you have to suffer through something like, let's say, hierarchical clustering using CPP-Trach or in Amber or another method, you know this can take not only a lot of your time, also a lot of your memory. And you usually don't have that available just like quite easily. My group has been developing tools to quantify separation between objects, like very general objects, essentially taking down the O(n)² problem to a linear scaling problem, both in time and in memory. For the particular case of conformations, we simply propose to use this MSD metric. So if you use MSD, essentially, you can use some extremely simple like algebraic manipulations to show that the composition of all the MSDs for any number of conformations in your simulation is going to scale linearly with the number of geometries in your system. So basically, you do have a new global way of quantifying the diversity of the conformations in your set essentially, and we will see this later, a linkage criterion for hierarchical clustering, and we can also use this for pathway analysis. And this is something I'm going to also like show in like a few slides by the end. We can use this trick not only to give you information on the global structure of your simulation, we can also use this to give information on the local structure inside your simulation. So let's say that you have multiple conformations, and this is a problem also mentioned before. How do you select most diverse and representative conformations from these like very long simulations? If you remove one and calculate the MSD of everything that is left, and you repeat this for every single conformation, basically, if these MSD values, what we call the complementary MSD, if they are like on the high end, on the higher end, this will indicate that the conformation that you remove on the other hand, if the conformation that you remove leads to a very reduced average MSD value, you are essentially identifying that as noise. So basically, with these two tricks, we not only have a global way to look at your simulation, we also have a way to give you local information, which you can use to detect outliers. Essentially, if you want to sample rare events, this might be a way that you might want to go about that. We can use this for sampling. We can use this to certify your data and essentially like prioritize different segments of your simulation for further analysis. So these two tricks are essentially the key, and like why MDANCE is different from like other packages that you have like maybe interact with to do MD analysis. Going back to the problem of how to pick a diverse set of conformations from your simulation. So if you use this trick of complementary similarity, we analyze multiple systems. We benchmark this with, to the right of the green line, you will see the results that you will get using CPP-Trash, like tools implemented in Amber. So not only are we finding more diverse conformations as measured by the RMSD on the y-axis, if you look at the time that it takes to get these results, if you use CPP-Trash, this can take you, if you use hierarchical clustering, is the one in the middle, on the order of hundreds of hours. If you use our tools, it will only take you just like a couple of hours to get equal, if not better quality results to approach this problem. As I was mentioning, I want to focus mostly in today's presentation talking about clustering. In MDANCE, we have hierarchical, we have density-based, we have a radial clustering. I want to focus on K-means, and actually, I think there was like a question on the chat on Zoom about like GaMD and K-means before. So this hopefully will like give like some extra insight for this. So I want to focus on K-means because first of all, it is extremely simple. It's incredibly popular. It also has a lot of problems. And I think that we have found ways to potentially overcome some of the issues with K-means, which can also be extended to other types of clustering. So first of all, exact K-means is an NP-hard problem. This means that essentially solving K-means exactly is as difficult as cracking like every single like credit card password that everyone has in the audience right now. However, approximated K-means is order n. Approximated K-means is incredibly efficient, hence why it is so popular in the MD community. The key problem with K-means, one of the key problems with K-means is how do we select the first initial K points? And that approximation is what makes K-means, like approximated K-means order n. By far, the most popular approach is just picking K random points. However, like we will definitely would like to do better than this. So we have to have like some sort of like heuristics selecting those like first K points. The most prevalent strategy is what is known as K-means plus plus. Basically, the key idea behind K-means plus plus is that you will pick a point, an initial point, now marked in green, and then your second guess should try to maximize the separation from that first guess. And then you keep repeating this process until you find those like initial K points. This is like the basis of what is known as vanilla K-means plus plus. There is a greedy version of this, which essentially does like a local optimization around each like potential candidate green point. And there is like a sort of like a hybrid approach implemented in CPP-Trash that essentially tries to convey the idea that your first K center should be as separated from each other as possible. Now, we propose a new way of doing this. Our K-means algorithm, this is like K-means n-ary natural initialization, or K-means n-ary, once again, like another cool acronym. In n-ary, we use two of the tools that I showed you before. First of all, We use the complementary similarity to identify high density regions in your data. So you don't have to sample points that are going to be outliers. So they're going to be very unlikely to be a centroid for your clusters. And second, we use diversity selection to find points that are well separated from each other. So basically you have your MD simulation, you calculate the complementary similarity. Once again, all of this is linear in time and memory. You calculate the complementary similarity, you identify high density regions, you focus only on those high density regions, and then in those ones, you are going to do a diversity selection, once again, like guaranteeing that you're going to be picking conformations that are highly different from each other, yet corresponding to sections of the data with like a lot of conformation around them. This is already publicly available. The paper was published in GCTC like early this year. Like feel free to drop by GitHub, like try it, let us know. It is also like MDANCE as a whole is also already available in MD analysis. So if you guys prefer to use MD analysis, that is already there. Also, like Paulo Arantes from Vita Nova Institute, he was kind enough to prepare a collab version of MDANCE. So there are multiple opportunities for you guys to try the software. So I show you essentially 4 different versions of K-mix. Now I need to try to convince you to use our version of K-mix. Like that, how do we evaluate the quality of different clustering algorithms? Well, there are different metrics there are different indicators that we can use that essentially tells you how good is your final clustering. For example, a very popular one in the MD community is the Kalinski-Harabas or the pseudo-F index that essentially measures how compact are going to be your clusters in the end. We have the silhouette score that essentially quantifies the separation between the clusters, and our HMSD, how compact are going to be the clusters, and the Davis-Baldwin index or the DBI that essentially quantifies the separation. All of this can give you some sort of information as to how good was your final clustering. Oh, sorry. And also, we are going to see that we can leverage these indices to also answer a very critical question. That is, how many clusters you have in your data. In short, we want to maximize the CHI and the silhouette score, and we want to minimize MSD and DBI. So now I'm going to walk you through different simulations that we tested, our method, and these other versions of K-means. First of all, if we focus on the pseudo-F results, and once again, bigger value means better, we see that the green line essentially corresponds to a CPP trash results, so not doing great. So the worst method of the 4 that we compare, while our method and the other K-means plus variants are essentially just doing giving you equivalent results. Now, while this type of plots allow you to compare different methods to do clustering, this in particular doesn't give you absolutely any information on what is the optimum number of clusters for your data. Because you see there is a strong bias towards low number of clusters. So anywhere that you will truncate this, essentially the method will tell you that the optimum value for K is the lowest value for K. So you should not be using the CHI or the pseudo-F to tell you how many clusters you have in your data. Can the silhouette score tell you something about that? Well, once again, it does a very good job at separating good clustering from bad clustering. And once again, the CPP trash version is not doing great. And once again, our method is doing equally, if not better, than the other K-means++ versions. However, once again, there is a strong bias towards low number of clusters in the silhouette score. The MSD essentially has the opposite behavior. Now there is a strong bias toward high number of clusters. Once again, you do see the tendency of CPP trash to not cluster the data in a very convincing way, if you want to. While once again, our method, the red line, performing better, if not equal, than the other K-means++. The one index that we found that actually give you information, valuable information about the optimal number for K for your data is the DBI. The DBI, we don't see an obvious bias towards low or high number of clusters. We also see these local variations, and essentially we propose 2 different mechanisms to identify critical points for K in your simulation. One is just looking at the absolute minimum value for DBI around your simulation, and 2 is finding the maximum value for the second derivative or the DBI values, which essentially tells you that a given particular K value tends to be more stable than the surrounding K values. So that's why you see two different marks, like the circle for the absolute DVA and the star for the maximum second derivative DVA. Now, I do want to, like, I haven't mentioned until this point, but if you look at the green, the blue, and the orange plots, you will see error bars. And in some cases, like pretty big error bars. This means that we took the same simulation, and we clustered them 3 different times using the same algorithm. And if you use the same algorithm multiple times, you will get different clusters. In some cases, dramatically different clusters. For example, even if you want to see the optimum value for K, let's say, look at the, if you use for the beta heptapeptide, the one on your left, if you use CPP trash, if you run that 3 times, in some cases, you will get that the optimum K is 5. In other cases, it's 30. So like you can get wildly different results. On the other hand, and to me, this is the biggest selling point to our method, there are no error bars on the red plots because our method is the only one of all of these different K-mean variants that is absolutely 100% robust and reproducible. So n-ary is not only fast and memory efficient because it is K-means, and once again, that is a capability like shared by every other possible K-means implementation, but it's the only one that is fully 100% deterministic. Like if you run your cluster analysis using this type of K-means, if you run it 1 million times, you are guaranteed to get the same results 1 million times. And you don't get like huge variation between like this, like over different like flavors of K-means, essentially. Okay. We tested this in the for the billing simulation. So we compared this to a recently proposed clustering method by Glenn Hockey, Martin McCulloch, and Heidi Clem. So they propose an absolutely beautiful JVMN clustering. We, with simply K-means, were able to reproduce essentially finding the same, like more important conformation that they found using this more elaborated method. One of my absolutely favorite toy models, so this like this also like folding, like a non-trivial case of folding. So this particular protein folds following 2 different pathways. We were able, so this is like what you will get with hierarchical clustering. Our K-means was also able to identify all the different metastable states along the 2 different protein folding pathways. Once again, at K-means costs. So you don't have to spend all the time and all the memory using hierarchical. I'm going to briefly go through this. Once you have finished your clustering, you want to find now what are the most relevant conformations inside each clustering. We propose like a new, in collaboration with a group of Alberto Perez, also UF, this like protein retrieval via integrative molecular ensembles, or PRIME. With PRIME, we essentially propose a way to, after you have done the clustering, how to identify representatives that are like more robust. Essentially, it boils down to, instead of focusing on your main cluster and finding the centroid of that, what if we also take information from the other clusters? And essentially, we not only try to find the centroid, but we also trying to find the conformations that are the furthest away from the other clusters. Long story short, once we tried this for different types of systems, like for low temperature, high temperature, replicate chain simulations, for like slightly more flexible proteins, and for protein-peptide interactions, you see these like blue columns essentially tell you the fraction of time that PRIME is finding conformations inside your simulation that are closer to experimental structures than just taking the average of the highest populated cluster. So once again, it gives you a very simple recipe to identify better conformations inside your clusters. Something, and I love like Yinglong's analogy with the like the whole if you are studying how a ligand is potentially binding or unbinding, you can see it's like it's trying to score a goal essentially in a game. So if you have a simulation, like a method like Gaussian acceleration, that allows you to sample and to see multiple binding and unbinding events, you might want to see if the mechanism of those binding and unbinding events is the same. So basically, now we are not talking about clustering a simulation. We're talking about clustering different simulations at the same time. There are multiple ways of doing this. This is an ongoing collaboration with Lydian Chong's group at Pittsburgh. So essentially, with Shine, this like sampling hierarchical intrinsic n-ary ensembles, we are also like leveraging the idea that we can compare multiple things at the same time incredibly quickly to now essentially cluster sets of different simulations and identify like intrinsic mechanisms in those simulations. The idea is very simple. You start with your ensemble of different like, potential pathways. We can do sampling, which is optional, but you can use our methods. It's like diversity oriented or like a representative oriented sampling to do this. We do the our MSD comparison between the different sets. With this, we build like a very simple matrix to then do hierarchical clustering on top of the different pathways. Basically, we have different metrics. It's like, don't worry too much about the formats, but more than happy to talk about those at the end if you want, to quantify how different conformations inside different simulations are connected to each other. And with this, once again, like we build a very simple matrix, a very efficient matrix, which we apply to, then we use like traditional hierarchical clustering on top of that. So the whole key of chain is essentially finding a better way, finding a better metric to quantify separation between different simulations. So far, we have applied this to two like simple toy models. First of all, like different like mechanisms for the alanine dipeptide. We're able to identify not only the two different mechanisms to go from one base into the other, but also the fraction of like times, like the correct fraction of times that alanine is like going from one base into the other. For a slightly more complicated example, different simulations of this like relatively more complicated system. Once again, we are able to not only identify that like there are like two dominant mechanisms, but also the actual fraction of the time that this like a binding like process is happening. We can also identify, once you have clustered all the different pathways, we can help you identifying the most representative pathway along the ones that you have already found. So once again, everything that you do for a single simulation, now we can do for multiple simulations. And once again, if you have a method like GaMD that allows you to do this like multiple times, a tool like Chain can like essentially with no overhead, you can get information on how are these different mechanisms being sampled and being explored. Basically, I hope that I convinced. I mean, like the one key line is the one at the end. Like there is a lot of stuff beyond RMSD. And like one of my goals in life is for people to stop using RMSD. If you compare multiple things at the same time, you do get a lot of information essentially for free, and you're able to analyze, you're able to do clustering in a better way, in a faster and more robust way. You're able to identify better cluster representative, and you now are able to even like cluster different simulations in a more efficient way. I want to thank my group. They are kind enough to post with like the one-page reference, which I absolutely love. I also wanted to mention very quickly, so I told you at the beginning, I had something for people doing small molecules. I just want to show you one key result that we just got in the group. If you're doing clustering for small molecules, there is a huge chance that you're using RDKit to do your clustering. If you're using RDKit and you want to cluster, say, on the order of 500,000 molecules, that will tell you essentially like close to 10 hours to do this. And you see like the prototypical parabolic dependency with the size of your set. We found a better way, a faster way to do clustering for small molecules, which we call Bedburg, and that is a time dependency that we have.
Key to the freshman lab.
Okay.
So you can
So
get the material.
it is
out of
linear
that.
in
You know,
time,
it's
linear
probably
in memory.
less.
The code is already available. And the one number that I want to like leave you with is that I think that we, as far as I know, we are the first team that has clustered 1 billion molecules. We not only did that, we did it in less than 5 hours. We like if you extrapolate the RDK plot, it will take you more than 6,000 years to do 1 billion molecules. Also, like we had to stop at 500,000 because 500,000 require 4 terabytes of memory to do. And so, and once again, like now we can do on the order of billions. So if you're doing like if you plan to do something, say with enamine, this might be like a very nice way to to clean and help you in the first step of your simulation. With that, thank you so much once again for the invitation. Happy to take questions.
Blake, please.
Yeah, that was an awesome talk. The development of your SHINE algorithm, very interesting. Like, could you elaborate on similarities and differences between that approach and say like a Markov state modeling approach? Is the amount of data that you would need to put together a pathway like a lot smaller than say using Markov state modeling? Because that typically takes a ton of data.
What we have been saying, so for these two cases, for the RNA peptide, the simulations were like relatively short. For the ADK, we had a lot more data like available to us. What we did is essentially clustering the pathways, trying to sample them. And we saw that even with like as little as 10% of your original data, we're able to reproduce these results. So in principle, you don't need a lot of data to identify data like this, like dominant mechanisms.
Would you potentially have like some sort of benchmarking mechanism or like a diagnostic way to determine like what a characteristic timescale would be necessary in order to stitch together those pathways?
We are currently not working in that. We should definitely be looking into that.
Yeah. Thank you.
Any more questions? Let's thank Ramon. And now we have a break, and we'd like to take a group photo. Maybe like for people on, I mean, on Zoom, we take like screenshots, but like for people who are here, like, can we maybe get onto the stage and take a photo? Yeah.
Really?
Maybe don't be almost the same. It's still a very brand new project.
So, yeah, I maybe can. Yeah, I just want I want to say for people on the Zoom, if you can please turn the video, so you can like take some screenshots like after the break. It's like a part of the group photo, right? Yeah, thank you. And yeah, welcome back everyone. I'll say, yeah, for our next talk, we have invited the speaker, Professor James Prestegard from University of Georgia. So Jim obtained a PhD in chemistry from Caltech. He spent 27 years in chemistry at Yale. So he went all the way from assistant professor to full professor. Then in 1998, Jim moved to the Complex Carbohydrate Research Center at UGA. Over there, yeah, apart from the professional duties, he also directs the MR facilities. He serves on the editorial board of many MR journals and also on the advisory and review panels. His group develops new MR methods. They also apply the MR to study the structure function of carbohydrates, proteins, and membranes. So today, yeah, he will talk to us about the conformational distributions of glycans on the glycoproteins use MR and the GaMD. Well, thanks so much for being here, Jim. Like with that, yeah, I'll turn it over to you. Thank you.
I should close some of these.
Let me give it a second.
Oops.
Yeah, we can.
That was. That's what you're. And then I need to share screen.
Maybe
Okay.
not yet. So that's on the screen here, but not on Zoom. Turn that on. So can everybody on Zoom see the slides? Yeah, all right, thank you.
Okay, how do I advance?
So you can just.
Okay, perfect. Okay. Okay, thank you. Thank you for that kind introduction. As this title indicates, I intend to talk about conformational distribution of glycans I'll start by telling you a little bit about that. I'll tell you a little bit about why and how we apply accelerated MD to looking at these. I'll warn you, that's not my area of expertise, but I present it so I can maybe get some comments and suggestions from the group during the day today. And then I'll tell you about how at the end of the day, we try to validate what we finally see by using experimental data. And that comes primarily from nuclear magnetic resonance. And I'll tell you just a little bit about what type of data we get from NMR. Okay, so let us try to proceed. First of all, why do we worry about glycans or carbohydrates or sugar residues? Well, the simple answer is that most of our proteins in our body are in fact not just collections of amino acids, they are glycosylated by covalent attachment of various sugar residues, and many of them quite complex ones. And of course, it's not just us. Pathogens that invade us are also covered with glycans. And this snapshot up here, this is from some work of one of my colleagues, Rob Woods at Georgia, early in the COVID pandemic, that looked at what are the glycans on the spike protein of the COVID virus doing, and what do they look like? All right, so the purple and green that you see there, those are snapshots of a long conventional MD run of the glycan residues on the surface of the protein. And this was done partly to try to identify regions where the amino acids were actually exposed and available for development of antibodies and so forth. But it gives you a snapshot of, why this might be important. And it also raises some questions. And, okay, this looks like they're really flailing around in space. Is that what they really do? How well is that distribution representing what really exists? And if we learn about what exists, what are they doing here? Do they have special functions? So that's sort of our objective. Now, we're not going to tackle anything as complicated as what I just showed you with the spike protein from COVID virus. I'm going to work on a selected domain. It's actually the N-terminal domain of a molecule called CCAM1. This is a cell surface human adhesion molecule, internal cell signaling molecule. It, like most of those proteins on the surfaces of your cells, is glycosylated. Even the single N-terminal domain has 3 glycosylation sites. They happen to be N-glycans. Let me see if I can get a pointer here. Is that showing up? Well, maybe not. Tried to get laser pointer, I'm not sure. Okay. So N-glycans are glycans that are attached to asparagine residues in the protein backbone. They all share these first 5 residues. There are 2 N-acetylglucosamine residues, and there are 3 mannose residues. This particular construct has 2 additional mannose residues out there. So it's actually called MAN5. Normal glycans here, I've depicted the 3 attachment sites here in conventional molecular depictions. But in a natural system, not all of those sites are equally occupied, and not all of the glycans on them are the same. Most of them are not limited to the 7 residues I show here, but have 9 or 10 residues with many other sugars. So it gets extremely complex. But we work with one of my other colleagues, Kelly Mormon, to express this construct in a way that has nearly full occupation of those three sites and has a very homogeneous set of Man5 residues. So it's something more feasible that we can look at. Now, ultimately, we're going to use NMR spectroscopy to look at the conformational properties of these glycans on the protein. And to do that efficiently, we've got to make use of protons, which are naturally magnetic and very high abundance, but we also have to add some other isotopes to help us resolve these complex systems. And so we'll add a carbon-13 residue biosynthetically in the expression medium. All right, and so that is something else that is important for what we actually do. Now, isotope labeling of proteins like this traditionally has been done by expressing proteins in bacterial cell cultures, which you can feed very simple substrates, glucose, ammonium chloride, nitrogen 15 labeled. We don't do that. We haven't done that for many years because it is very difficult to do in mammalian cells that properly glycosylate proteins. They don't make use of things like ammonium chloride. They like to be fed amino acids that are already labeled, and that gets very expensive if you try to uniformly label. But it turns out there's some very simple ways to get enough labels in there to progress and validate structures, providing you know something about the structures. And for proteins, at least these days, we have structures for essentially all known proteins from AlphaFold. And it's a perfectly acceptable way to start. All right, and so our job is much simpler, very sparse labeling. And it's accomplished by simply putting in some extra glucose labeled only at one position, C1 glucose. This is relatively inexpensive. A liter of mammalian cell culture, you're going to put in about 5 grams of this. It costs about $100 a gram. So it's quite feasible to do this. It ends up in all sugar residues. I've listed just some of them, but the anomeric C1 carbon and the sugars of all those gets labeled. Not only that, this goes down through glycolysis to pyruvic acid, and that label ends up in the methyl group. And then there are single enzyme steps to get to acetyl-CoA, so all acetyl groups get labeled, and a single enzyme step to go to alanine. So all alanines get labeled, all in the methyl group. So it turns out to be very important because for NMR, they are one of the easiest things to see, very high sensitivity. So this is very feasible, relatively easy work to do. Now, getting conformers of glycans that we can test by collecting NMR data, we do by accelerated molecular dynamics. That's why I'm here talking about this. And just to stress why this is important, I mean, years ago, we ran conventional MD. All right, so this is a distance map of a distance between one of those labeled N-acetyl methyl groups and some residues on the surface of the protein. All right, and this starts out at pretty close contacts, about 4 angstroms or so. And this is a long trajectory for conventional MD. It's a microsecond. It takes us a week or so to run this on our computer systems. And you notice what you see some rattling around, but about halfway through, you see a transition to a much more extended structure. All right, and looking at this, you'd be extremely tempted to say, you know, okay, I've found the dominant structure in this system, and you'd be just totally wrong, okay? And you turn over and do accelerated MD, this is what you see. And you see that this starts out, and it samples things up around 12 angstroms, just like this, but it continually keeps going back. to those very close contacts. So this is a very different picture. It's important that we learn something from that. Now, what do we do? And as I said, I'm not an expert in this, but I'll tell you a little bit about what we do just for maybe comments further on into the day. What we'd like to do, of course, it's hard for us to, handle those, 100,000 different snapshots in an MD trajectory and look at the NMR behavior of all those. We'd much rather have 5 or 10 most representative structures that we can easily work with. And that's what we'd like to pull out. What are the most populated states that we see? All right. And so that's what we try to do. And of course, we use various versions of accelerated MD. And in our case, we've chosen to excite those 3 glycans individually. We take the final trajectories that come out. We've got to try to now convert those scattered populations back into something that approaches our normal Boltzmann distributions at room temperature in our solutions. And our approach, we actually cluster those structures based on the similarity of glycan structures. And then we recalculate probabilities for those structures using some of the cumulative approximations that have been brought out in the past, and try to recover those populations. And then we will extract some representative structure from each of those clusters, either by any number of criteria. Very often we'll just pick the minimum energy structure. All right, so that's our plan. We do this just for interest of some people may be interested in this. A lot of what we do is based on MATLAB scripts, which is something we find very, very useful and convenient, easy to use. It turns out that unless you're a really super programmer in something like C++, you can't beat the efficiencies of CPM, which has just these optimized functions, higher order functions. So it turns out to be quite useful, particularly for inexperienced users. But this is just shows you the graphic interface for one of these scripts. It is based on our calculating an agglomerative hierarchical cluster tree in terms of this Z function, which you see this program is actually going to read in that function. It's some data about your actual trajectory. And then you have some variables. There's a cutoff and there's a depth program. The cutoff, you saw one of those cluster trees in Ramon's presentation, and you notice the legs separating various members in that tree have certain heights. All right, that's what this cutoff is regulating. And so you can adjust this to sort of retrieve a certain number of clusters that investigate these repeatedly if you want. This, I'm just showing, this list goes down. There are actually about 28 clusters in what I've done here. And then we calculate these probabilities, and this is just showing a plot of the probabilities of these various clusters. So we'll pick out, maybe 5 of these, which, is manageable for us to go back and look at in some detail. Now, NMR, and what do we do? So, this is a picture of our molecule down here, and there are those glycans. If you see most structural NMR, it's based on the nuclear Oberhauser effect, which is a proton-proton experiment, which has very steep distance dependence, 1/r⁶. It's very sensitive for things that are a few angstroms apart. Beyond about 4 or 5 angstroms, you don't see anything. Not very useful if we're trying to look at glycans sitting out here. right? Which can be tens of angstroms away from the surface, right? So we need something that's different. And we go back and we rely on some long range data that come from things called residual dipolar couplings and pseudo contact shifts. Now in this case, they both depend on us introducing something that has a very high anisotropic magnetic susceptibility. And in our case, lanthanide. And all right, because if I have that in there, it does two things. One is put it in a magnetic field, you induce a big dipole moment in it. And that is a second order field that now extends out about 40 angstroms at a useful level. And so I see perturbations in my chemical shifts. Those are called pseudo contact shifts. That's one variable. The other is you got an anisotropic induced magnetic moment in a magnetic field. Different orientations have different energies of interaction, and our molecules no longer tumble in sample space isotropically, they have preferred directions. And the minute you do that, you see dipole-dipole interactions between pairs of nuclei, like my carbon-13 and my proton. That's directly attached. If you had two bar magnets that represented those two sites and you pulled them in and out, you realize there's a distance dependent, it's 1/r cubed. And if I do this with them, you realize that there's an angle dependence as well. And that's geometric information that tells us a lot about where those glycans are. So these equations represent that. Here in the pseudo contact shift, down here, you see there's a 1/R cubed. That's these long distances, very long range. And you see these angular dependences here. You see some variables I haven't talked too much about. The delta chi's, those are the anisotropic susceptibilities. I don't know those a priori, all right? And I've got to determine them. So the strategy here is, I know this protein structure. Okay, AlphaFold tells me what that is. I know where all my alanines are. All right, and so I can go up and I can use pseudo-contact shifts, all of those, to actually determine what these delta chi values are. And once I know those, I can calculate, knowing distances and angles for sites in my glycans, what the observed RDCs and pseudo-contact shifts should be. All right, so that's the strategy for doing a validation. Now there's some caveats. Well, let me first just show you what pseudo-contact shifts look like. All right, so this plot, this is a proton bicarbon-13. It's actually for methyl groups in our system. NMR two-dimensional spectrum. They're superimposed two different acquisitions. One with a diamagnetic metal in my protein, and one with a paramagnetic metal. The diamagnetic ones don't have any pseudo-contact shifts. That's just like the normal spectrum we look at. They're in black. And then red is with a terbium ion. All right, and you see the chemical shifts change. Now, remember, these are out 30, 40 angstroms away. I'm looking at a proton directly bonded to a carbon-13 methyl. All right, they're an angstrom, 1.1 angstroms apart, right? 30 angstroms away. All right, the R is 30 angstroms, right? So the values for the pseudo-contact shift in terms of proton or carbon-13 are virtually identical. That means that a diagonal line connects all those pairs. All right, so I pair up all these spots for all my alanines in this case. Down here, we've also put in some labeled valine, which has two methyls on the side chain. All right, and so they're a little further upfield, they're up here. But our pseudo-contact shifts is just measuring the length of those vectors. All right, so this is an extremely simple experiment. Two-dimensional NMR takes 1 or 2 hours to collect each one of these. And the analysis is simply sitting down with a ruler and drawing line. All right, so This is a very straightforward way to get data, except there are always caveats to this. One is I have to know which alanine is which in terms of sequence.
Hey Google.
So I can assign them.
Hey Google.
Pardon me? Yeah,
Stop.
okay. We use a program coming from a lot of Sign SLP to do this. I don't need to go into that very well. Once I do, I get those anisotropic susceptibilities, and this is plotted for a particular value of chemical shift, 0.07. And here I superimpose my molecule. You see, glycans are going to be mostly in this upfield shifted region, but some of them actually go in the opposite direction when they get up into this blue area. So it's actually pretty widespread. This is the picture now, again, superposition of two of these HSQC two-dimensional experiments, black and red for diamagnetic and paramagnetic again. And the connecting lines are the pseudo-contact shifts, but these are now the anomeric carbons on all of my sugar residues. These are assignable by because they're chemically different. So most of these you can classify into sugar type, linkage type, and so forth. There's superposition because we've got three chemically identical glycans. But you notice that the minute I put in a paramagnetic ion, these tend to be differentiated, all right? And I can resolve here are the second glucanac anomerics in this case, all right? The three are almost resolved, but you notice the resolution goes up dramatically when I put the paramagnetic ion in. And so this is the way we collect data on that. This is for the acetyl methyls on our glucanacs. All right, there are 6 of those. You can see there's 1, 2, 3, 4, 5, 6 red residues that are resolved. We can get lots of very useful data. We can also collect residual dipolar couplings. Now this is more angle dependent, right? Because it's just the carbon-13, the directly bonded protons. The distance between those is pretty much fixed. It's a bond length. So it's primarily angle dependent. We actually add a third dimension to our NMR experiments. We don't transform it, but we collect it as a sinusoidal wave to fit that. And down here is an example of experimental, and this is pseudo-contact shifts for these acetyl, this particular acetyl methyl, and here's the in the H1, here's the RDCs over here, and you see predicted and experiment for a particular frame that I've just picked out of this trajectory to take a look at. All right, so this, these look fairly clean, but the range should point out for the anomeric proton, the range of those couplings is actually about -25 to +50 hertz. Okay, so these have to be fairly small, but yeah, the range is important. So they're giving us quite a good picture of how distinct these are. Now, this back to our clustering and looking at most probable structures. And so I've just picked out now from our accelerated MD run, the minimum energy structure in the 3 most, I don't know if these are, 2 of these are the 2 most probable structures, and then there's 1 that's in the other top 5. And down here you see experiments, and here you see predictions for those particular structures. Some of these agree fairly well. I mean, you can follow these across. Experiment -0.16, you see a couple of these are very, very close. Here's a very small residual dipolar coupling. Some of these are very small, and so forth. down here, here's another residual dipolar coupling experiment is 7. At least one of these clusters fits that fairly well. But you start looking at these in detail and you see that so far, looking at these, we've not found a single structure that fits all of the data well. And that's probably not a surprise. These are fairly dynamic systems, and they are properly represented as an average of these structures. n-ary, by the way, averages variables over time scale from picoseconds to milliseconds. And so it actually fits the accelerated MD time scales fairly well. And that is another valuable asset. I want to show one more snapshot. You'll notice that these structures, and these are trimmed back. I'm only showing you the first 2 N-acetylglucosamine structures. There are 5 more mannose residues out here. And you notice these first 2 residues, they are quite similar in all of these high probability structures. Most of the variation is in what I'm not showing you, which are those mannose residues. But you notice the similarity is sort of surprising, that so many of these frames end up having something very similar. You notice these acetyl groups, which are yellow, are pointing in sort of the same direction here, 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 and here. And you notice they're close to the protein surface. Well, we can go back and take a look at one of our short range experiments, nuclear Oberhauser effect, down the range 4 angstroms or so. And this is depicted here. And you notice this acetyl methyl is actually in contact, close contact with hydrophobic groups. All right, a leucine and a histidine. All right, these are little hydrophobic pockets. All right. And this is a repeated observation that these glycans, what they're doing is they're finding little hydrophobic pockets on the surface of the protein. And it may be important to cover those up, prevent interaction with other molecules that they shouldn't be interacting with. They may contribute to stability. Those are things we don't completely understand at this point, but this is a start at understanding. And that's essentially my message for today. It's a start at understanding, combination of experiment and some computational predictions. This is credits to the people who have actually done this work, and in particular, Jinan and Yinglong have been very helpful in overcoming some of the difficulties in running these trajectories. One, which I neglected to mention as I went through, is that water models make a huge difference for things like glycans. And tip 3P does not work very well. And so what I showed you is all run with tip 5P, which is just another one which actually works somewhat better. And it's just one more message of, where there's still work to be done and why we don't have complete understanding of these systems. And just a final one, a credit to how we do our n-ary work these days. It's done through a cooperative venture called NAM that's supported by the National Science Foundation that has n-ary facilities at the University of Wisconsin and University of Georgia. But all of the computation and the capture of all the data and how it's made available to the public is done through computer resources at the University of Connecticut Health Sciences. And with that, I thank you for your attention.
Time for questions. Please.
I'll just keep asking questions. Jim, great talk. And I'm glad you brought up the point about the tip 5P, because I was trying to remember, like I used to work with glycan when Rob was just first using it, except came out of Pete Riley's lab at Iowa State. And I don't know if, has he updated Glycam since '06?
And he's been updating that ever since I've been at Georgia.
Okay.
And they've not released the final version yet.
Okay, gotcha.
But you know, it is a little tricky. And you know, I say, well, you know, tip 5P works better, but the fact is that the Glycam force field is optimized to work with tip 3P, right?
Sure, yeah.
Right. So, you know, which is the better route to improving this? Maybe it's just simply improving the Glycan force field.
Well, yeah. But I was gonna ask too, because I wasn't familiar if Glycam was compatible with OPC. That's Alex Onufriev's water model. That's compatible with the guy like the amber force fields, but that might actually allow you to get a little bit more performance. And I don't know how well it compares to tip 5P with accuracy, but might be something to consider.
Which do you know which one you're referring to?
It's an OPC.
Oh,
abbreviation.
OPC. Yeah, okay.
It's shipped with amber as well.
Some of this comparison has been done. There's sort of a review by I think it's Sergei Samsonov, which went through several of these water models on a particular type of glycan. And tip 5P is one of the two that seemed to work best in what he looked at. You know, but I think that's sort of an ongoing investigation, and it's much more complicated than, you know, it seems on the surface, because you're changing one part in a complicated, you know, system which spans a much greater set of variables.
Right, carbohydrate force field for tricky.
Yeah, yes. Yeah.
One question on Zoom, is there any particular reason to cluster based on manners instead of the protein backbone fluctuations?
That's a good question. You know, we clustered on individual glycans, mostly because our thought was that if I cluster on much larger subsets, that I'm going to have a lot of noise to deal with. And, you know, the thought was that a targeted system may be somewhat better. But again, you know, this is an area of research, and I welcome comments and suggestions.
But let's thank Jim. And our next speaker would be Professor Shirley N. From UC Davis. So Shirley obtained a PhD with Eric Dove in chemical physics at Stanford. And then she had a postdoc with Henrik Chem and Romeo Mario at UCSD. Shirley started her lab in chemical engineering at UC Davis in 2022. Shirley develops new enhanced sampling methods for MD and also apply these methods to study important biophysical phenomena. So Shirley has received a number of awards, including the Chancellor's Outstanding Postdoc Award at UCSD, and also the ACS Physical Chemistry Division Young Investigator Award in 2021. Now today, she will talk to us about bridging the timescales in protein submissions using parallelized GaMD or ParGaMD. So with that, Shirley, please.
Oh, yeah. Yeah, thank you for the kind introduction, Yinglong, and for the invitation. So today I'll be talking about an ongoing enhanced sampling method development that is happening in my lab right now. And I titled the slide as Bridging Timescales in Protein Simulations Using Parallelized Gaussian Accelerated Molecular Dynamics, because as everyone knows in this audience, there is an inherent timescale barrier between simulations and biological processes, because for MD simulations or molecular dynamic simulations, we have to use femtosecond time steps because we're limited by the fastest motions in the system, such as vibration of bonds. And as everyone knows, biological or biophysical processes that we're interested in are on the order of milliseconds or longer. And so enhanced sampling method developers like Yinglong and myself and other people, we're basically trying to bridge the timescale barrier between simulations and biological processes so that we can actually obtain useful properties like thermodynamic and kinetic properties in a computationally tractable period of time. And so I think for this audience, I don't really need to go into what are molecular dynamic simulations, so I will skip that. But as Jim was mentioning about glycans and SARS-CoV-2 spike protein, I also did a bit of work on that during my postdoc days, but I'll just skip this slide for to be on time. And then also MD simulations are applied in various areas, including material science, drug discovery, for investigating important biological processes. But I will mostly focus on the enhanced sampling methods for MD simulations, which try to bridge the timescale barrier between simulations and biological processes. So as I already mentioned, we have this inherent timescale barrier challenge in the MD simulation community. And besides the having to use femtosecond time steps to simulate up to microseconds, milliseconds, or longer, most of the time when we actually run these MD simulations, we're often stuck in these low energy metastable states for a long period of time. And so when we run conventional MD simulations, we have to wait for a long period of time for the system to actually go over the energy barrier and then explore other conformations, or for us to actually see conformational changes for proteins, for instance. And so there are many researchers who are actually working in this field, in this enhanced sampling method development field for MD simulations, Gaussian accelerated molecular dynamics, or GaMD. I don't think I need to really talk about that for this audience, since Yinglong gave a really nice introduction for this. But including Yinglong and other people, there are several researchers who are developing biasing methods for MD simulations, which add a bias to the system so that we can actually sample these metastable states that are separated by high energy barriers, but by adding a bias to the system, we're able to lower these energy barriers so that we can sample from one state to the next, and the transitions more efficiently so that we can sample all this in a computationally tractable period of time. And so besides GaMD, there are also methods like metadynamics, which also adds a biasing potential to the system, but still slightly different. And also replica exchange, which instead of adding a bias to the system, it actually changes the temperature of the system so that we're able to sample conformations much more easily. And besides the biasing sampling methods, there are also path sampling methods like the weighted ensemble method, which essentially, instead of running one long single simulation, it runs many short simulations in parallel so that we can efficiently sample the associated thermodynamic and kinetic properties directly from the simulation. And so I will talk about this in great detail because essentially what Parallelized GaMD does, which is what we're trying to develop right now, is to try to use this weighted ensemble method framework to run parallel GaMD simulations so that we can actually reduce the wall time significantly for these GaMD simulations and sample thermodynamic properties more efficiently from our simulations. And I guess the last category of enhanced sampling methods that I should mention is there are also methods that try to divide up the entire configuration space into small macrostates or bins or small volume elements of the configuration space, and then tries to sample the associated thermodynamic and kinetic properties with that. And the weight ensemble also falls into that category, as well as Markov state models as well. And so I'll briefly introduce about the weight ensemble method, because I don't think a lot of people in this audience knows about it. So, and I think it'll be very useful for many researchers, because not only can you apply this for MD simulations, you can also apply it for Brownian dynamics, coarse grain simulations, etc. It's a very, very general method that was actually developed by Gary Huber and Sang-Tae Kim back in 1996, I think. And so I'll explain about this method in detail. So let's say you have this simple configuration space that you would like to sample thoroughly. And so using the weighted ensemble method, we have to define a few parameters beforehand. So first, we need to define bins, or small volume elements that divide up the configuration space. And so for simplicity, we'll have 3 bins that are dividing up this simple configuration space. And so from here, we also have to set the progress coordinates, which are also called as collective variables or order parameters, depending on which terminology or jargon you specifically use in your research area. But these progress coordinates essentially tries to measure the progress going from a reactant state to a product state. And so for protein simulations, it's usually RMSD, dihedral angle, distance, etc., depending on the system. And in this case, we'll just set X to be our progress coordinate for this simple system. And then we also have to set the simulation length tau for these short simulations, because here, instead of running one long single simulation, we're going to run many short simulations. So in practice, we usually set this to be from 10 picoseconds to 100 picoseconds, depending on the size of the system. For our SARS-CoV-2 spike system, we set the simulation length to be about 100 picoseconds. And then for the smallest system, like alanine dipeptide, for instance, we usually set it to be 10 picoseconds. And then finally, we'll also set the target number of short simulations per bin, and I'll refer to short simulations as walkers from now on to be concise. And the rule for the weighted ensemble method is that we need to maintain this set target number of short simulations per bin for each of the visited states. And so here, we'll set that to be 2, and we're just starting off at this very initial state. And so we'll end up with just 2 walkers in this initial state. And these walkers will also carry a probability or weight associated with them. And they also have to add up to 1, because probabilities, if you sum them up, they have to add up to 1. And so each walker will carry a weight of 0.5. And so from here, we'll run the simulation for tau amount of time. And here you'll see that one of the walkers has transitioned from the first bin to the second bin. And since one of the rules for the WE method is that we need to maintain this target number of walkers per bin, what we're going to do is we're actually going to split this walker into 2, and the same goes for this one. And so we end up with these 4 walkers with also the probabilities and weights splitted as well. And so from here, we're going to run the simulation again for tau amount of time for all these 4 walkers. And in this case, we see one of the walkers from the second bin has transitioned back into the first bin, which is likely to happen because here we're in a high energy region. And so it's likely that we'll also see a transition falling back into its lower energy region. And so from here, we're going to carry out this process or maintaining the target number of walkers per bin, or that's called resampling. And for, whoops, where's the mouse? Oh, here, okay. And so for this walker, we're going to split it into 2 as we've done in our previous step. And in this case, we have 3 walkers, but we want to maintain 2. And so what we're going to do is we're actually going to merge these 2 into 1 so that we end up with 2 walkers in this first bin. And the goal is that we want to save our computational costs for these easily sampled areas, and we don't want to waste our computational resources and effort in sampling this low energy state. Whereas by splitting the walkers in these high energy, rarely sampled regions, we're focusing our computational costs in sampling these rare events. And so from here, we'll run the simulation again. And here you can see one of the walkers from the second bin has transitioned into the third bin. And you can see easily, even in this simple example, that without resampling or splitting this walker into two, we wouldn't have been able to efficiently transition from this first bin all the way to the third bin. And so you can see the how resampling is giving efficiency for the WE simulation. And so this, I think I'm gonna skip over, but this just goes into the resampling technique and how it's doing the resampling within each bin. And we do this in a statistically unbiased way. And so one thing to note before I get into ParGaMD is that actually the default original Huber and Kim method in the WE method, the resampling technique actually ends up yielding walkers that are not equal to each other or not equal to the mean weight of the walkers in each bin. And so if you run this for a long period of time, we'll have some variance and statistical errors associated with this as a result. And so in order to mitigate this issue, we can actually easily set the walkers to have equal ideal weights, just like I had in the previous slide where I just added up these weights and then divided it by 2 and assigned those equal ideal weights to each of these walkers. And that way we can actually minimize variance and statistical errors. And so that's a really easy fix to reduce errors for the WE simulations. And so actually, one of the first projects that I did since starting my independent career at UC Davis is I had one undergrad researcher. His name is Dennis. He graduated just this past June, and he actually implemented this equal rate resampler for WE simulations that can be directly used for WESPA, which is the most commonly used WE simulation engine in our community. And he was actually able to show that with this equal rate resampler, when we actually compared it to the standard default WE engine, we're actually able to sample the high energy regions more quickly than the standard resampler, which is remarkable. This is for an analytical toy model system. And then we're actually also able to sample the correct rate constants. This is for a simple NaCl system in explicit solvent with also lower error bars than the standard resampler. And so with this easy fix, we were able to demonstrate this for 2 simple systems. And this was recently published in Journal of Chemical Physics this past July. And so now that I've explained about the WE method in detail, now I'm going to talk about the ongoing method development for parallelized GaMD or ParGaMD. And so I don't think I need to explain about this in detail since Yinglong already gave a nice introduction about GaMD. But one nice thing that I should actually mention about GaMD is that this is a collective variable free method, which there aren't many of because most of these enhanced sampling methods in our community, we actually need to know what are the optimal collective variables beforehand a priori, which is actually pretty difficult for new unfamiliar systems. So GaMD is a really good method to actually explore new systems in a CV-free way. So I often use this a lot for my other application projects in our lab. And so I think it's implemented in more programs than just AMBER, NAMD, OpenMM. We've mostly used GaMD implemented in AMBER because AMBER has one of the, or not if the fastest MD GPU engine as of today, or it might be OpenMM. I'm not sure. Someone could correct me if I'm wrong, but anyway, so we've been using Amber with GaMD a lot for our application projects within the lab, but we've noticed that it's actually not well parallelized. So Amber does have a function where you can use more than one GPU to accelerate the simulations, but as the Amber documentation notes, it's really not parallelizable for GPUs that are newer than 2016. And so we noticed that when we run like GaMD or LiGaMD on really big biologically relevant systems, such as alpha-synuclein with interacting with small molecule inhibitors, for instance, it would take about at least 2 weeks on a single GPU to run it up to a microsecond. And since we need to run replicate simulations and we actually had a lot of small molecule inhibitors to test, it took basically months to get the data. And so seeing this for one of my grad students' application projects, I thought, oh, I mean, if it's going to take weeks to reach, microsecond timescale, 1 or 2 weeks, why don't we actually try to parallelize it using the WE framework? And so that's how we were motivated to come up with this new method called parallelized Gaussian accelerated molecular dynamics. And so currently 3 students are working on this. So Siddharth is leading this project along with Celeste, who are my 2 graduate students. They're 3rd years in the chemical engineering department. And also Maxon is one of the undergraduate researchers in our lab helping us out with this project. And so basically the idea is very simple. First, we're going to be running a short conventional GaMD simulation to finalize the boost potential parameters, because if you actually get into the detail of the GaMD algorithm, as explained in this diagram, it's also on Yinglong's lab website as well. It actually takes some time to finalize the boost potential parameters, around like 2 nanoseconds by default. And so after we run that short conventional GaMD to finalize the boost potential parameters, we can actually use the finalized parameters to run parallelized GaMD with the WE framework, or WESPA to be more specific. And as you've seen for the WE method, we actually need at least 1 CV to actually use the WE framework to run these parallel GaMD simulations. So that could be a caveat. So we do need to know at least 1 CV that would be relevant to sample the system of interest. And this has been worked on over the summer by Siddharth, because this project is mainly funded by the Duke Center for HIV Structural Biology, because they would like to also accelerate MD simulations of very large systems, which the center is trying to study. And so Siddharth was working on the ParGaMD implementation, and it's actually available for use on his GitHub website. So anyone can actually start using it now. And so now I'm going to talk about some preliminary results that we've gotten so far. We're still working on to develop and test the method, but so first we actually tried to run ParGaMD on the alpha-synuclein small molecule inhibitor system that Celeste is mainly working on. It's about 200,000 to 300,000 atom system. So it is a considerably big system. And so we wanted to actually show with the ParGaMD, we would have close to linear scaling with the number of GPUs. And indeed, you can see this is the relative speed on n number of GPUs compared to 1 GPU. And you can see it has close to theoretically perfect linear scaling, which is really, really great. And also the speed as well, you can see we can get up to almost close to 1.4 microseconds a day, which is really, really great compared to the 1 GPU speed, which is very, very low. And so we were able to actually test this on the Texas Advanced Computing Center's resources, the Frontera resources, because there aren't that many supercomputers in which we can actually test up to more than like 16 GPUs at a time, but we were able to test it on up to 48 GPUs. So we were able to get this really nice linear scaling. And I should thank Frontera Pathways allocation for the resources for this work. And so the second preliminary result that we got is that we actually tested this our ParGaMD method on Chignolin, which is a really well-studied system. Chignolin is a 10 residue mini protein that's cited in many enhanced sampling method papers like Yinglong's and myself and other people as well. So we wanted to test ParGaMD on Chignolin in explicit solvent. And so you can see the free energy landscape evolution at 100 nanoseconds, 200, 300, 400, and 500. And you can see the metastable states that are between the folded region, which is basically the region in which RMSD is less than 1, 0 to 1. And then the folded state is considered to be states where RMSD is 4 or higher, but you can see the intermediate metastable states throughout the simulation, which is really great. And then this was the very final energy landscape that we got from running this for 1,000 iterations, or about a little less than 8 microseconds, which took about 2 days using 48 GPUs. And so we also wanted to compare our ParGaMD performance with respect to the control WE simulation, as well as the GaMD simulation as well. And so when we compared it with our weighted ensemble simulation, in which in both of these cases, we basically started off with the same one basis state that started off in the folded state after running GaMD for just 2 nanoseconds, so that we have the finalized boost potential parameters. And you can see a significant difference in the free energy landscapes between ParGaMD and the weighted ensemble method. And this actually shows that with the accelerated boost potential from the GaMD portion, we're able to sample the free energy landscape way more efficiently than just the conventional weighted ensemble simulation. And they both had the exact same conditions of like one basis state, and they were run for the same amount of molecular time, and you can see a significant difference between WE and ParGaMD. And this is what you get after you run it for about 1,000 iterations as well. I just plotted it in 2 different energy scales so that the metastable states are shown more clearly in this scale, whereas this one is more favorable for the weighted ensemble simulation. But regardless, you can see like even if we run it for a long time for the weighted ensemble simulation, it's far from being converged. And so finally, we also wanted to oh, whoops, I think this is a typo, sorry. This should have been 500 nanoseconds. We also wanted to compare our ParGaMD results along with the GaMD results, because one of the hypotheses that I had for ParGaMD is that since we're using WE to propagate these trajectories, and it's propagating the trajectories based on the chosen CVs, I hypothesized that ParGaMD is actually going to be more efficient than GaMD due to the added CV-based propagation. And so here you can actually see there's actually a significant difference in the free energy landscapes. Although the coverage is about the same, you can see that GaMD needs to run a bit more than 500 nanoseconds to get these intermediate metastable states well sampled. And so you can see that with ParGaMD, there's also a significant performance improvement over just conventional GaMD as well, which is really great. And so these were the Chignolin simulation statistics for all these different 3 simulations. You can see that with ParGaMD, if we use 48 GPUs, we get about 38 times faster on ParGaMD with 48 GPUs compared to our just one GPU conventional GaMD simulation. It's not exactly linear scaling, but still we're able to sample it a lot faster if we actually have these available GPU resources. And you can see that it's a bit slower than WE, but still I think the speed is pretty comparable between ParGaMD and WE. And then finally, we also wanted to see whether we're able to sample more transitions in these three simulations. And so we kept track of the number of folded to unfolded transitions and also the reverse transitions as well within the 500 nanosecond simulation time. And you can see that ParGaMD, it was able to sample these transitions more frequently than WE and GaMD, and which makes sense because if you see the free energy landscapes, ParGaMD was significantly closer to being a converged free energy landscape compared to the GaMD and WE simulations. And so with that, I would like to conclude that ParGaMD is a promising new enhanced sampling method that would mean less molecular time than GaMD due to additionally having WE sampling along the chosen CVs, which is really great. And then, of course, it also needs less molecular time than WE because we're running GaMD instead of conventional WE to propagate the trajectory. So the boost potential is really helping a lot. And so these are the QR codes for my lab website and also for the ParGaMD GitHub that Siddharth is maintaining. And I would like to acknowledge my funding resources, the Duke Center for HIV Structural Biology, that was the main funder for this project, and also my computing resources for running all these simulations, and also Celeste and Dennis and Maxon as well for working on this. Thank you.
The next talk is Shirley.
Great talk, Shirley. I have one question, like what kind of GPU models used during the study?
What kind
What models, like NVIDIA GPU models used during the study?
GPUs? Oh yeah, so for the scaling, we actually used RTX, like I think 4000. They're very, very old GPUs, but they're the only GPUs that were there on Frontera, and Frontera is the only one where we can actually test significantly large number of GPUs. So yeah, we use very old GPUs. But I expect like if we use slightly newer GPUs like V100, which is still old today, but like A100 or newer, then we would probably get a significant speed up. So yeah.
More questions? Please.
Sorry, I keep asking questions. What CV did you choose for the Chignalin?
Oh yeah, so for Chignalin, we used
Was just radius of gyration or?
Yeah, we use both of these CVs, RMSD and radius of gyration, because this is a very well-studied system. So in the future, I think we've exhausted most of our analysis for Chignalin. So we're going to also test it on the alpha-synuclein small molecule inhibitor system, in which we use LiGaMD. And for that, we just use center of mass distance between the two as the CV. So I'll test that, and also probably more general protein systems. And we want to test with like more general CVs so that we can actually use ParGaMD for other systems as well.
And then in comparison to like your vanilla GaMD and the WestPA, maybe to go ahead a couple slides. Yeah. So is there any experimental data on what the energy barrier is for folding to unfolding for Chignalin? Like, I guess which scale is more accurate? Is like the 12 kcals per mole more accurate?
The 5 kcal per mole, yeah, is more accurate.
It's more accurate.
Yeah.
Okay, that makes sense.
Yeah, and we're going to extend the GaMD run to like almost 8 microseconds so that we have a fair comparison between all three. So yeah. Yeah, but it's a very well-studied system, so we can get the reference free energy landscapes from other literature as well. Yeah.
All right, thank you, Shirley. It's very nice. All right, our final talk would be by Professor Pengfei from Loyola University Chicago. I see Pengfei over there. So yeah, he would give
Yes.
a talk over Zoom. Pengfei obtained a PhD in chemistry with the Kennedy Mass at Michigan State, and then he had a postdoc with Sharon Hermes-Schiffer at UIUC and Yale. Pengfei started his lab at Loyola in 2020. His lab develops metal ion models. They also work on simulations and molecular design for the metalloproteins. Pengfei has published more than 37 papers with thousands of citations. Since 2015, Pengfei is also a developer for MBA. And today, yeah, he would talk to us about running GaMD for customized regions in OpenMM. So without further ado, yeah, Pengfei, please go ahead.
Thank you, Yinglong. Yeah, thank you for the invitation and the introduction. So today I'm going to like share some like learning I have got like in the past like maybe about the past a year. Yeah, also share some findings like some code like example files with everyone. So the title of my talk is Running Gaussian Accelerate Molecular Dynamics for Customized Regions in OpenMM. Can everyone see the screen I'm sharing?
Yes, it's
okay, great, cool. So yeah, first I will give some introduction like about OpenMM and yeah, about like GaMD in OpenMM, something like that. And then I will talk about the integrator and then talk about the force field. So basically molecular dynamics, you can consider molecular dynamics as like force fields plus integrator. And then I will show like simulation procedure for an like example, and which is like a metal ion binding to a host system, and then finally we can see like results like about we use GaMD in OpenMM, like, and then we can do some simulations and we can compare the results with classical MD, and finally it's acknowledgement. So about this talk, the code and example files are available on GitHub, and so it's like on my GitHub like folder there is like a project called Customized Region GaMD, and inside it you can find the example files. The example files like the pump topology and equilibrium structure you can find in this folder, and you can also find the OpenMM code to run classical MD and the OpenMM code to run GaMD. So with the like customized regions, yeah. So the version of software I use is OpenMM 8 and ParMed like 3.4.3. So well, different version or combination may not work. So you may need to adjust the code to make it work if you use a different combination of the software. And also, like, I want to make a clear declaration here is like the code and the file deliver in hope that they are useful, like without any warranty. Yeah. So, and first I want to give a quick introduction about OpenMM. OpenMM is I think a very user-friendly software if you want to make customized integrator and customized force field. And so basically is like, you can think molecular dynamics as force field plus integrator. And then you can play around with like either elements and make your customized like molecular dynamics. And OpenMM has been developed in the past, especially in the past decade. And So for example, the following are 3 references about OpenMM. The first one is about OpenMM 4 and and then the second one is about OpenMM 7. And last one is the one recently published in this year. Earlier this year is about OpenMM 8. And in OpenMM 8, they supported also machine learning potentials. Yeah. So which is make OpenMM is a very powerful, I mean, and like molecular dynamics engine to run. And also it would be very easy if you want to make customized force field or customized integrator in OpenMM. And this is the diagram of classes in OpenMM 6.0. I haven't found an updated plot about the OpenMM 8.0 yet, but I think is the major diagrams do not change. Yeah, so it's basically you have like different kind of classes, and there is API layer classes, like for example, also there is app layer classes, like or app layer classes from files. So they are marked with different colors here, and here like it's showing like the relationship about each classes. And so basically you want to create the central classes is like you want to create a simulation class, and you can like create a system first, like based on force field or based on pump topology file, and this system will contain forces. And then based on the system, you will create a simulation, and also like for example, you need an integrator to for your simulation, and finally you can run your simulation, and then it can it can create contact. What contact has a state. Like state contains positions, velocity, energy, and forces, and so on. So is here, this diagram will be helpful when you do your coding, yeah, because it tells you like clearly about like which kind of classes you needed to create which kind of classes. Yeah. And this picture is from the website. Like you can find the link here. Okay, that is a like very brief introduction about OpenMM. And if you are interested, you can also dig into their website. Yeah, so have like extensive resources about OpenMM and example files, and also manual. Yeah. So about GaMD in OpenMM, actually Yinglong's group developed the code for GaMD in OpenMM about 2 years ago, and they published in Physics and Biomedicine. And today I'm going to show another like implementation of GaMD in OpenMM, which might be a more straightforward way in general. So what I refer to is the AMD integrator in OpenMM like already. So this code, the accelerated AMD is already in OpenMM, and it was written by Peter Eastman. And if you are interested, for example, if you use Miniconda to install the OpenMM, then you can find this code under this path. So in the side packages, OpenMM and AMD.py. And inside this code, it has 3 classes. So AMD integrator and AMD force group integrator and dual AMD integrator. So the first class is AMD integrator for the total potential energy. And then the second class is for the AMD integrator for a specific force group. For example, non-bonding, if you want to accelerate non-bonding interactions only, so you should choose the second class. Choose this integrator to run your AMD simulations. And the third one is dual AMD integrator. So basically you can boost the total potential energy and plus a specific like force group, for example, non-bonded like potential energy. Yeah. So by following this framework, and also by refer to like other code, for example, the code from LG Martin on GitHub, and also trying to be consistent with Yinglong's ligand MD work, GaMD work. So I I coded, created a file, and you can find this file like in the GitHub as well. So it's like in this folder. So you can find that file here. Yeah, OpenMM_GaMD.py. Yeah. Inside it also has has 3 integrators. These 3 integrators are trying to be consistent with the AMD integrator in the OpenMM. Yeah. So basically today we are going to show like one example about using the dual GaMD integrator is trying to simulate a system, a metal ion binding to a small molecule, to the 18-6 molecule. So, and inside for this system is we are trying to boost the total potential energy plus the non-bonding interaction energy between the metal ion with the whole environment. The whole environment means not only the host molecule, but also the water like solvent surrounding. Yeah, but the code can be used to simulate other kind of system with customized regions. So, for example, if you want to boost the interaction energy between between a ligand and with only a part between, for example, between the ligand and only a part of protein, you can follow a similar strategy. Yeah. So there is an interaction group in OpenMM, interaction group function in OpenMM. You can like create any customized interaction group by yourself. Yeah. And then you can use a dual GaMD integrator or GaMD force integrator to boost your potential, either in a single boost way or in a dual boost way. Yeah. Okay, the system I'm showing you is like cesium binding in 18-6, like this molecule. So for the example files, I'm using AM1 BCC charges plus GAF for the 18-6 molecule, and I'm using SPC water. The periodic boundary box size is like 40 × 40 × 35 angstrom. And inside the system, I only have 1 cesium ion and 1 like chloride counter ion to make our life easier. Yeah, and then the cesium and the chloride parameter are from the parameters we developed before, and which can reproduce the hydrogen free energy of these 2 ions well. Yeah. And then I used like 2 boosts. Like one boost is the ion environment interaction energy, and the other boost is the remaining potential energy. This is trying to be consistent with like Yinglong's LigaMD work. Yeah. So now I will take some time to talk about the non-bonding force in OpenMM. As we talked before, this is about integrator. Now I'm talking about the force fields. Yeah. So in OpenMM, when dealing with non-bonding interactions, there are 2 kind of functions you can use. One is the non-bonding force function, which is by default for the electrostatics and Lennard-Jones interactions. And there is another function called customized non-bonding force. And inside this one, you can set interaction groups between any parts of the system. Yeah. So, and for the non-bonding force and the custom non-bonding forces, there is one thing we need to pay attention, which is the cutoff consistency. Yeah, there are different cutoff approaches. So in the non-bonding force, in this function, there are in total 6 options you can choose. Cutoff non-periodic and cutoff periodic, EWAR, PME, and LG PME, and no cutoff. Yeah. So for the cutoff non-periodic and cutoff periodic, actually they are using a reaction field model, trying to reaction field model is to deal with the atom within the cutoff. Yeah. with a, like assuming a uniform solvent like outside the cutoff. Yeah. So for example, if you are using a cutoff equal to 10 angstrom, like we are using either cutoff non-periodic approach or cutoff periodic approach, we are using non-bounded forces. Like this function, so you are assuming like anything outside the cutoff. For example, if you are using 10 angstrom cutoff, you are assuming anything outside the 10 angstrom is like a solvent with dielectric constant equal to a specific number. If water, then it will be dielectric constant will be approximately equal to like 78, something like that. Yeah, but the atoms within the cutoff, you need like, yeah, there is a specific like function for the non-bounded like electrostatic interactions. However, if you are using like customized non-bounded forces, there is only 3 options nowadays. In the future, I'm not sure whether they will add more options. For example, add like E-word like implementation as well, or some other approaches. But currently, it only supported 3 different kind of cutoff approaches: cutoff non-periodic, cutoff periodic, and no cutoff. So cutoff non-periodic and cutoff periodic, the difference between the two is like for cutoff periodic, it will treat the system like it will like see the periodic boundary box. So it will basically take that into consideration. But if you're using cutoff non-periodic, you will not consider the periodic boundary box into consideration. Yeah. But if you are using, for example, a cutoff values for cutoff non-periodic or cutoff periodic, when you are using customized non-bounded force, by default, it assigns a hard cutoff. Yeah, by default, it assigns a hard cutoff, not a reaction field model. Yeah, so which means if you want to keep consistent between the non-bounded force and customized non-bounded force, you need to like code the reaction field model into the customized non-bounded force energy functions when you created the customized non-bounded force. Yeah. So yeah, further details you can check like this web page is a conversation by Alex and Peter Eastman. Yeah. So and also you can find like in the coding you can find I did such kind of implementation. Yeah, if you check the code. So I did such kind of implementation when created the customized non-bounded force. Yeah, following like Alex, like his code. Yeah. So okay, that is about the implementation. So in the example is I used a cutoff periodic for the non-bounded force, and also I use cutoff periodic for the customized non-bounded force. Yeah. So the simulation procedure is shown below. First, I did structure preparation. So the initial structure of 18-6 is from graphic anti-chamber. Graphic anti-chamber is available in Professor Junmei Wang's his webpage. So Junmei is a developer of general amber force field. Now also he developed it like a cool like website. So for example, if you go to like his website and then go to the first one, like you can sign in as a guest and anti-chamber, then you can select graphic anti-chamber and you can like draw any molecule here. Yeah. Draw any molecules here. Yeah. So that, and then there are like other functions you can follow. Yeah. Feel free to play around with this. Yeah. This is a very cool website. So I use it to create like the host molecule structure, and then I added caesium through Gauss wheel, and then I solvated the molecule using a SPC water box, and then added chloride ion to neutralize the system. And afterwards, I used PPI-GaMD Lacuda to do like minimization, MBT heating, and MPT equilibration to get an equilibrium structure. And the equilibrium structure is you can find it inside this folder. Yeah. So this is the Parm topology, and this is the equilibrium structure after MVT, after MPT. Yeah. So And afterwards, I did both classical MD simulations using OpenMM and also LigaMD. I called a LigaMD Duo. Yeah, this is a customized region, like GaMD in OpenMM. And then I run this simulation using OpenMM. So you can see actually the speed is, this one is a little bit, is kind of slower. Yeah, it's maybe about only 60% of the speed of the classical MD. So this is because inside this one, you created a customized non-bounding force. Yeah. So it's basically, when you are running the two systems, you are running two like kind of non-bounded interactions in parallel. Yeah. So that's why the like speed is kind of slower. Yeah. And here instead, this one is for the implementation is I turned off the ion environmental interaction for the non-bounding force, but for the customized non-bounding force, I only have ion environment interactions. Yeah. So in this way, I separated it to generate an independent force group from like the remaining potential. So, and for each of the classical MD and the LigaMD Duo simulations, so it's like I run is 3 replicas for the production, and each replica lasts like 15 nanosecond simulation. And so and the snapshots was saved each 1 picosecond. So giving like 150,000 snapshots for each of the like classical MD simulation or LigaMD Duo simulation. Yeah. So this number of snapshots actually we'll see in the results is not good enough for publication. Here I'm just showing you like for the demo purposes. Yeah. So like, but if you want to like, for example, publish papers, I would like, I think Yinglong suggested that like you better to have like 1 million like snapshots for like rereading. Yeah. So here is under 1 million, but just for like demonstration purposes. Yeah. Okay. So this is the results. Yeah, this is the results of on the x-axis is the distance between cesium and the center of the 6 oxygen atoms. So we have 6 oxygen atoms on the host atom, and then the center of it, if we like measure the distance between the cesium and the center of it, and then we calculate the potential mean force along this like reaction coordinates, and then we plot the data here. There are 3 data set I'm plotting here. One is classical MD, which is shown in black. Yeah, and one is GaMD related, which is shown in red, and one is GaMD without reveating. It's showing in green. So we can see that here the profiles are not very smooth. Especially for classical MD and the GaMD reweighted, it's not very smooth. This is because the number of snapshots we use are not a huge data set. Yeah. So if you have huge data set, then you will have like a more smooth curve. Yeah. And we can see for the GaMD reweighted one, it resembles the classical MD results pretty well actually. Yeah. So they are like within 1 angstrom. Yeah. 1 kcal per mole, within 1 kcal per mole for the potential mean force. Yeah. So which is pretty good. Yeah. And for the GaMD no reweighted, and we can see which is the green curve. And if we compare it with the classical MD, which is the black curve, we can see encouraging things actually. So the GaMD no reweighted one it's actually more smooth. It's more smooth than a classical MD. So, which means for sampling is more evenly distributed. Yeah, and also for the GaMD no reweight, is like we'll get a lower free energy. So it resembles the well and the peak agree pretty well with the like classical MD, but in general, its free energy curve is lower than the classical MD. So which means like GaMD is really showing like a boost. It's really showing like enhanced sampling in this way. Yeah. So. And yeah, this is something like I want to share with you. And if you are interested, you can download the code and trying to play around by yourself, and you can test other systems. Yeah. Okay. So finally is like a quick advertisement about my group. So my group is going to recruit at least one PhD student, who started in fall 2025. Yeah. And assistantship will be provided. If you are interested, or you know someone who is interested, you can like let him or her know. The admission reviews begin in December 1st and close by April 1st. So, and also if anyone is interested, you can like check my group's website. So it's like, yeah, it's like here is like showing the current members and also our group news and photos. And yeah, we have the opening is, yeah, this is here. So yeah, and a very quick introduction about the university I'm in is called Loyola University Chicago. So it's located in the northern part of Chicago, and it's just by the Michigan Lake. Very beautiful campus. Yeah. So finally, I want to acknowledge first, I want to acknowledge Dr. Yinglong Miao for his invitation and also for his help along the way. And also, I want to acknowledge the startup funds from Loyola University Chicago, and also the computational resources, which is from San Diego Supercomputer Center, which is provided through the Access platform, which is funded by NSF. So I would like to thank you for your attention.
Yeah, I have time for questions.
Sure.
Any questions? And also on the Zoom.
I can send you the link through the chat about the code on GitHub. So anyone, yeah.
Yeah, that's great. We have one question here. Pengfei,
Sure.
please. Like Blake or Shirley.
Okay, I have a quick question. So, with this customized like GaMD boost potential, can you also supposed to start our seminar soon? So please wrap it up. Yeah. Okay. Yeah, mine's a quick question. Can you also just boost like just certain atoms, like for instance, like if we're just interested in like alanine peptide, like bi and side dihedral angles, can we just choose those regions with this program?
Oh yeah. That's a great question. So basically, in OpenMM, you can create any like customized forces, not limited to non-bonded forces. You can also customize bonded forces, bond angle torsion, and like these ones. And you can select the regions, any specific regions through the interaction, like for the non-bonded forces through the interaction group, like that function. Yeah. So for example, if you want to just select the region like inside a protein, you can do that. Yeah, no problem.
All right, thanks so much, Pengfei. Yeah, we're having another seminar here, so stop it here. And we'll be back at 1 o'clock, about 50 minutes. Yeah, thank you.
Thank you.
home. Just a lot of, the more you think about it, the more questions you come up with. For example, we're running these are Gaussian MD. If you cluster after that, do you just get rid of any benefit of having Gaussian distribution? So we're recalculating probabilities after the clustering.
Yeah, we still have a couple of minutes. Then for people on the Zoom, we'll come back. I think you can take our time now to connect to the UNC cloud accounts if you get the resources. Otherwise, you may download the files for the tutorials. So you can get the tutorials to your workstations and make sure you have them ready. Maybe you can, next would be Victor. So he will go through the GaMD tutorial, maybe in about 1 minute. Take your time.
Sorry, I'm without the instructions, I actually don't know. Let me think.
Is your outlet work?
Yeah, before.
They're not bad.
So someone in front of you.
I'll try. Yeah. It's a level.
That's what I thought.
Oh, wait. So that nerd file. With your username. Gosh, yeah, this one. Can you copy it to your?
Like done before. That's probably better.
Which key file?
Oh, and then
You can ask them. Sorry. So which folder is that key file?
Welcome everyone.
Okay.
Now we'll be starting the hands-on section for the GaMD tutorial. So yeah, the first section we're going to look at how to run the normal GaMD simulations, and I think you should have this file, like both the remote users and also the local users. So I will not talk about the introduction because we've had a lot about GaMD, and I'll just go to the logistic part of the tutorial. So here this is our system, is a alanine dipeptide, and we want to run GaMD simulation on alanine dipeptide. So for the logistic purposes, we have these schematics that is talking about the GaMD, and when you log in, I'm still going to provide a very brief intro, introduction on how to log in, but let me just describe the schematic. So when you log in, we have the GaMD. Inside the GaMD folder, we have two folders, simulation folder and also the analysis folder. So in the simulation folder actually contain all the input files to run your GaMD simulation. Whereas in the analysis folder, you have all the input files to run some of the basic analysis, to look at to run the PMF, also to get some of the, for example, the diagonal angles, the psi and the phi angles. So this is for the logistic purpose. So basically, you don't need to copy anything. You just need to, for now, this is the schematic we're going to follow. So we can just go through the tutorial now. So basically for UNC local users. So this is the instruction. I didn't like. For this, if you want to log in, you have already been given a key file. So you just do SSH I and then the key file with your username and then the GaMD workshop.rnc.unc.edu. So you should be able to log in. So I'll be able to log in, and if I do pwd, so this is my home directory. I'm already there. Here there is no file there. If I do ls, I don't have any file. So what we're going to do is to so now we're going to just like copy in this shared data file. We have the GaMD that contain all the input files. So for the remote user, what we're going to do is just like we're going to copy this folder into your work directory. If you're able to log in, like, has anyone be able to log into the UNC cloud there? So what I would do cp -r.
Copy minus the third comment. But in Linux.
So you just type this command, and this should be able to copy the GaMD folder from shared data directory into your work directory or your home folder. Now I should. So now, like if you have any trouble doing that, you can raise up your hand so that we can. So the next thing we're going to do to actually run the simulation, we're going to cd into this folder, simulation. Based on this, right? So we are entering this command now. And then if you want to look at the files, you can just do this. And then you see all the files. So basically the files we're going to use to run these simulations are like, they are this file. For the remote user, you have, so if you click on the remote user, we have to click on we have to click on this file. This gamd.star.bz. So when you click on this file, you automatically download the file you're going to run. That's to run the simulation. That's for the people without the UNC account, the remote users. So basically, you have to do the same thing, just archive this file, then you get a GaMD folder, then you go into the simulation folder, and then you're ready to start the simulations.
Victor, there seems a typo in the talk command. So we have uppercase GMD.
Oh, yeah. So, yeah, so you will get this file, GaMD dot. So basically, it's just like GaMD, GAMD, capitalized, then dot star dot bz. I want to be sure if everyone has gone to this stage, like, so that we are ready to run the simulation. Okay.
What procedure are you guys gonna use? This should be the handbook. What do you have?
To run the simulation, we just submit this, like we do this S batch run this script. So we just say S batch. So that's how we submit the script. So you can just copy and paste the script and so now I've submitted the jobs. If you want to see whether the job is running, you can do this. SQU, just like SKU, something like that. And then
Yeah.
I think some people have submitted jobs already. So yeah, So while we wait for the job running, we're going to like talk about some of the input files because we're going to wait for the job to finish running. I think we're going to wait for like 10 to 12 minutes to run the jobs. So to talk about the input files, you're going to get some of these files. Like it's going to start generating some files, and some of those files are the trajectory files, the MDANCE file that contain the simulation output files, like it contains the potential energy, the system energy, the kinetic energy, and the likes. And then you have your trajectory file. Then you have the GaMD restart, the GaMD1 restart file that contain both the coordinates and the velocity information. Then the GaMD log file is a very important log file because it contains all the boost energy that be generated. So you can basically like compute, you can use it for reweighting, because if you don't get this file, there is no way you can do the reweighting. Then another one is the GaMD restarts.dat file that stores the potential statistics, that's the average, and also the standard deviation of the simulation. And the MD info file will tell you like how long you still need to wait for the simulation to complete. And like, so you have these files being generated. So if you still want to see all those. So yeah, I think I still need to wait a little bit because of the jobs that have been submitted to see this file generated. So I'll just quickly go to what is in the MD.in.file, because this is actually the file we use to run the simulation, the equilibration. So it's the same with conventional, with the normal conventional, like if you want to run conventional MD, this file is the same. The only difference is what we highlighted here. So these are the minimal set of parameters that we include in the file. So you have the I-GaMD. I-GaMD is that is 3. If it's 1, you are running like you are adding just the total boost potential to the system. If I-GaMD is 2, that shows that you are doing you are boosting the diagrams as well. If it is 3, then you are boosting both the total potential energy and the diagram angle. So the I-GaMD when it's 3 is more efficient. So basically you can sample a lot of conformations of the protein. And for the high-res GaMD, if it's equals to 0, it means like we are starting a new simulation, like it's a new equilibration. And for these parameters, I will just explain how we actually get the values for these parameters. So let's take, for example, if you want to do like, if you want to know how long you're going to run the equilibration for in order to get the desired endpoint. So for alanine, the peptide is about 20 atoms, but when you add water and some other things, you get to about 11,500. So the total number of systems, the number of atoms, the maximum number of atoms in this system is around 11,500. Then the NTWX100 is just like the steps to write the trajectory. At every 100 steps, you write the trajectory. Then the NCPLT is for the number of proteins, which is 20 in this case. But because like the system is very small, so I'm not going to go with the number NTWPLT, I will go with the total number of atoms, the Nmax, which is like, so this will enable me to actually run the simulation, because if it's 20, that would be too low, and then you get like maybe like an error or something. So what you do is, for this, I've talked about the high GaMD 3. For the NTIF, then there's this NTIF, which is the, there's a default for NTIF. So for the NTIF in the GaMD parameter set here, so how we got that is, that would be 4 times of the total number of atoms in your system. So here we have 11,550. If you multiply it by 4, you get something like this. But you need to approximate it and make it an whole number. So it's like 50,000. Again, for MTCMD, MTCMD is the number of conventional steps to get the potential statistics. Like in GaMD simulation, you first run like a short conventional MD before you go to the equilibration proper. So that'll be 10 times NTIF. So when you multiply this 10 times 50,000, you get 500,000. So that's how we arrive at this value. But for the sake of this, because if we are to go through all this, actually we have to run the equilibration for around 5 nanoseconds. But for the sake of time, we just reduce these values to the default values and change this because that will take a lot of time, and change this n-slim, which actually show the total number of time we have to run the simulation to about maybe 3 nanoseconds, I think. So here we are just, for this simulation we are running it just for 3 nanoseconds, not 5 nanoseconds. For a normal simulation, based on the calculation here, it should be for 5 nanoseconds, like in this case. So basically, we should run it for 5 nanoseconds in this case. But because for testing reason or for practice reason, we are just running it for a very short time. So I think I can go back to see my jobs have been.
I'm guessing how to load in.
I don't know, it's not.
So I'm guessing this is connected to the remote.
Oh, yeah, I think that's wait, it's taking long to generate results somehow.
Is it still in the queue or is the job running?
Yeah, I think it's still in the queue somehow.
Looks like it's running.
Yeah, it takes some time to warm up.
Yeah.
Oh, okay.
But has anybody be able to generate any results?
I have a question. Why are we, like in the run, why are we starting with equilibration right away?
Yeah, so we are starting with the equilibration because like for GaMD simulation, you have to get a very well equilibrated system. So normally we run a very short conventional MD before we start GaMD simulation. That's basically to make the system to be well equilibrated for GaMD equilibration. Yeah.
But like, why don't we minimize before we equilibrate, for instance?
I think that has already been done during the conventional MD, the minimization. You have minimized the system, done a little bit of short equilibration. But because you want to do GaMD simulation, then you have to, even in GaMD simulation, there is a short conventional MD. There is another level of short conventional MD equilibration before you proceed to GaMD simulation equilibration proper. So I think that's because like you would imagine we are adding boost potential, the system can probably blow up. So you need to have a very stable system and well equilibrated system.
How do you choose the sigma?
Oh yeah, thank you for the question. So the sigma OP and the sigma OD, they are the default settings. So yeah, for GaMD simulation is 6.0, so it has been tested and is the default parameter. Yeah.
Like the width of the.
Oh, so that's like the magnitude of boost potential you are adding to the system. So if the sigma OP, if you reduce this sigma for other people who are going to come, they are going to talk about the sigma OP and the sigma OD because these are actually the two parameters we adjust to optimize our system to know whether the system is for example, for selective GaMD, you might need to like change the sigma OP to about maybe 2 or something to see whether it's going to fit into what you actually want to see. So if you want to see like, let's say dissociation, and then you want to see a very quick dissociation, you need to optimize these two parameters. So you selectively boost the ligand or the peptide or the protein, in whatever case you want to do. Then for you to actually see the faster dissociation, then you need to reduce the sigma OP.
Oh, reduce it, okay.
Right. So I think yeah. Has anybody been able to generate any results?
Well, the calculation is done, but I don't know what I got.
Oh, okay. Then okay, if the calculation is done, then you can
Yeah,
just.
you're gonna have to walk me
Okay.
through what I got.
Okay. Yeah, Oh, yeah, great. So, so these are the files. So if you look at the tutorial, like don't put the tutorial documents, you will see this contain the trajectory files, and this, if you open the MDANCE file, it contain the some of the maybe like the potential energy or like you see all this potential energy, the bonded and the non-bonded interaction and everything. So basically, this information are very important. So you're going to need them to proceed to production. So I think the most important file is the GaMD restart.dat file. If you open this GaMD, yeah, exactly. So basically, this contains the average and the standard deviation of the boost potential, that we added. So
Where is it for?
yeah, so it's going to have this is like maybe like the initial potential energy. Then this is the standard deviation. This is the average and this is the standard.
And what is it? It's a column. What does it correspond to?
So like if you open the GaMD1.log file. Yeah.
Oh, this one?
Yeah. So basically we have this file, the unboosted potential energy.
Oh.
Yeah, so we have the unboosted dihedral energy, and these two are the boost energy potential and the boost energy dihedral.
Okay.
So basically it takes this column, and also like this column and this column. And then it computes the average.
Okay. Yeah.
So I think someone has got the results. I don't know. I think I'm getting there, but again, there's no result generated for now. I think it's very slow. Has anyone also got any other results? Like be able to finish the calibration?
Oh, mine's running right now, I'm not done yet.
Okay,
Yeah.
it's running.
I have jobs on the workstation. It says like 1 or 2 minutes left.
Mine's also still running.
Okay.
You guys use long live at all?
Yeah, long live.
You use it?
Yeah. I know that it's going to take this slow. That's fine.
Victor, can we submit your job? That's what Rob suggested.
Oh, okay. Yeah, it's running now. Okay. Yeah, so I can quickly go back and explain what these files are again. So you will get like these files. MDANCE files actually contain all the system information. So if I do VI, MD1. So basically it's going to be generating all these files, like the number of steps and also the temperature, the pressure, and the potential energy and the rest. So for that's just what the MD file is about. Then for the GaMD log file, I think the most important file is this GaMD1.log file. Oh, it hasn't started generating. So I think I just have to wait for the other ones to generate. But basically the GaMD file log file actually contain the boost potential energy that are added and also the dihedral boost potential, the dihedral boost potential energy that are added. So the GaMD log1 file is very important, and also the GaMD restart.dat files. Those two files are very, very important. Okay. They have to be generated. So the restart file is just like it contains both the coordinates and the velocity information for it at every 100 steps of the trajectory that is being generated. Yeah. So I think the other things are just like conventional MD. The only difference is just the GaMD restart.dat and the GaMD1.log1. Any further question? Yeah, so for the remote user without the UNC local account, so there's a script. So, but you need to first solve the AMBER part. Like, so if you're a remote user, you don't have access to the UNC account. Just the way you used to run simulation on your laptop, you need to first solve the AMBER part. Once you do that, then you are free to just run the script, the bash script that is there. But I think the bash script that is there actually shows like you are in the home folder. You have to be in the home folder, the home directory rather. Then you just do like make the script executable by doing chmod +x, and then you just run the script. As for I think the instruction is up here. Yeah, this is the instruction for the remote users without the UNC account.
Well, while we're waiting for these to finish, I was curious, like, because not all systems are created equal. So are there some general rules of thumb for the amount of simulation time you should run for equilibration for like the conventional MD and the equilibration of the GaMD cycles as well?
Right. So based on this calculation, then we can. So that was why I said, like, for practice purpose, we decided to reduce the time. In this case, that's the end slim to about 150,000 or 1.5 million. But in the, like, if you want to run the simulation proper, you need to do the calculation based on the number of atoms in your systems.
Sure, that's one way to do it. But I mean, like, even so, you guys work on membrane proteins quite a bit too, and like a membrane takes much longer to equilibrate than a soluble protein. So yeah, I guess that's the question I'm asking is like, normally when we're running a membrane simulation, I wouldn't start collecting data for any type of analysis for at least 50 nanoseconds. So is that typically what you guys do?
Exactly.
Yes.
Yes.
As well?
Yeah. So particularly with the membrane system, like the G protein with the receptor with the G protein. So in that case, we can run the equilibration for about 70 nanoseconds. Yeah. That's all. But because this is a very small system, the equilibration is just within 3 nanoseconds. And for practice purpose, it's just 3 nanoseconds.
Are you talking about just during setup prior to running?
Right, yeah, right.
Because there also tends to be a period where the boost potential vary
high
after you started. We find this, we have to sort of empirically plot from the energy log file to find out when that distribution is stopped. Rising, it tends to be fairly long. I don't know if that's common.
Right, so I think like when we are doing reweighting, we put into consideration so if the boost potential is more than 50 kilocalories per mole, we exclude that because it's too high. So we put that in our reweighting consideration. So yeah, to get the accurate payment after like we completed the simulations.
Yeah, this is our.
You can do CAD-MD info and you can see how much time is left.
Yeah, I can do it. Yes. I think maybe my simulation stopped or something. I don't know. But something is. Oh yeah, I think it's there. 2.1.
You can check how many jobs are still in the queue. Okay, great. I think most are running.
Right.
No.
Takes time. So I think I get in there.
Yeah. This one.
Yeah.
Okay.
I use a long loop.
Yeah,
it's much faster, right?
Yeah.
Yeah, I'm using long live and general queues, like usually like if you just submit one job, it's like it's very fast.
Yeah.
Started in production.
So it's shorter. It's
Oh, okay.
it's missing 1,000 minus 1.
Okay, okay. I haven't.
So
After the calibration.
Yeah.
Just energy starts to produce.
What are you looking at? Like right away?
Yeah, already I did.
I'll let you know.
Temperature.
One as long as we're starting to
Accelerate.
like
there's nothing.
it's like the type of creation usually have some called an equilibrium.
Yeah.
Is this something that you think about or you it doesn't matter?
That's a great question.
Yeah, thanks.
To some extent, adding a boost is like increasing the temperature. We have a faster energy. But I mean, in our approach, like we added a potential boost. But I don't think we change the kinetic energy. You can actually look at the MDANCE file. Because it will output the temperature values. You can plot that for that.
Yeah.
Well, because I'm not actually checking.
But I would say that should not.
After I run the calibration and like get my files, look at the log file and everything, what's
So,
the next
right.
step? Do we run
Yeah,
the files right away?
Yeah. So you run the production, once you get the.
But then in the production as batch file, I don't see empty.m
Yeah.
at all. Like, what is that? Where to
The
here?
net effect is
Like, I see all these files, but what about this? Like, the empty
for estimation.
file?
The
Where,
system
when
have
did you
just,
see that?
just like what we have. They are weakening that measures, achieving that on forces. And it's like given from the
Oh,
rest
so
or.
this was just in the calibration. Okay, thank you.
Solid temperature. Yeah, solid temperature.
Almost done.
Yes.
Sure. You guys know about Pengfei's GaMD implementation of them?
Oh, no, much. Yeah, so we have,
Oh, but they're both. the same.
I mean, it talked to me maybe
Yeah, okay,
several
cool,
weeks
thank
ago. So he was working on that, yeah.
Okay, gotcha. I mean,
Yeah.
it makes sense what he did.
You also mentioned that to me, right? Did we? I think somebody else, maybe before, about kind of like a selective GMD in OpenMM.
Yeah, I mean, it's almost a way of doing like a REST2 implementation,
Yeah.
so to speak. But okay, I'm just gonna email you. That's the question. I'm just curious, like what the similarities and differences would be in the implementation of between what you guys
There was
did
one
and what he did.
errors, so they stopped all the jobs.
Oh, okay.
Okay.
In OpenMM, it's much easier to select like a group of atoms for like adding boost or changing the force. So it may be just a fast, easier for the implementation.
Yeah, because you guys basically generated the GaMD runner
Yeah.
executable from
Yeah.
scratch.
And we use the TI functions to help with that.
Okay.
Select like a group of atoms.
This is done now. Yeah, I think we're done now. Then we can just proceed with the production the same way we did. So we ran the equilibration. We can do as batch and run production dot PDS. So I think this is running. Then for this, we're going to get like it started generating. So we're still going to wait for some time to generate, but you can see like the files are being generated, MD2, GaMD2, that's for production, and also the MD2 out, and then the MD2 restart, that's fine. So let's look at what is actually in the GaMD, the production file, that's the GaMD restart.in, the input file we go run the production. And for this file, so we set the NTS, so it's going to read the coordinates and the velocity from the last restart file, and also with the restart parameter. But I think we should just focus on here. So for the GaMD parameter, again, all other input files are the same with the conventional MD, except for this. In this case, we don't need to add the NTCMD, the NTCMD, NTEB, and all this value will be 0 because we are not adding, we are not doing the GaMD equilibration. We only keep this NTEB So basically, this is the parameter set that is unique to GaMD simulation. The I-GaMD remain the same. The high-res GaMD in this case is 1. And also like but the NT-CMD, NTEB, and the NT-CMD prep and NTEB prep will be 0 except for the NT, which is 50,000. So, and then we should get files like MD2 out, MD2.nc, GaMD2.log file, and also, yeah, I think those are, and also the GaMD2.restart file. So I think for the sake of time, we can just go to the analysis part while we wait for this to get completed to see how we're going to, because we are not going to use any of these files for analysis because you're not going to see anything, like we're not going to get any result. So now, then I will go into the analysis folder and so I have actually done this simulation so I ran like 30 nanoseconds in 3 replicates. So totaling to 19 nanosecond simulations. And then I've actually also I've done the analysis. I've got the psi, I call it psi-hall.stat file. Those are the diagrams for actually doing some analysis with the alanine dipeptide, and also the phi-hall.stat. So these 2 files, and also I coordinate all the log files from those 3 replicas, and I call them GaMD-all.log file. So we need the GaMD-all.log file, the phi-all.stat, and also the psi-all.stat to actually run the analysis and get the free energy profile for this. So I think we don't need to do the analysis, but if you're curious, like if you look into the analysis folder, you can just do CPP charge and then select the dipeptide molecules and for the analysis. Yeah, so like this is just a basic CPP charge script to get maybe the diagrams and the rest. But we don't need to do that because the results are already in the analysis folder. We just need to now calculate the PMF. So we are done with the simulations. We have all the results, the diagrams and the GaMD all log file. So now we want to do the reweighting. We want to get the free energy. We want to get the PMF, which is the potential mean force. So I really don't need to talk about this. Another thing we're going to do here is also calculate the harmonicity that actually tells us the extent to which our simulation actually follow a Gaussian distribution. So basically, after you have run like your GaMD simulation, and then you want to do some kind of checks and balances, and then you want to be confident that like it seems my simulation is good, then you can actually also plot the harmonicity and see the distribution of the harmonicity and match it up to that of the original PMF you got from this result. So here we're going to calculate the 1D PMF, and also we're going to calculate the 2D PMF. We're going to also do the 1D harmonicity. We're going to plot that, and also we're going to plot the 2D harmonicity to see whether our simulation actually follow a Gaussian distribution profile. So here we have some scripts that to run that. So the first script for UNC users is this run 1D PMF.pdf. So we need to submit, we just do sbatch and submit the script, and then we get some result. So for that, let's just run that now. So we can do sbatch run 1D PMF.pdf. So if we run the script, then we should get some result. Yeah, so it's generating some results, and I think I can just go through those results, like what we are likely to get. So for UNC, this is the script for submitting the jobs, like is highlighted in black. You can see like you just take, in this case we want to know for psi angles, and we want to calculate the PMF for the psi angles, and then we take two files, the GaMD log file and also the psi dot that, and then we have this script that is actually doing the job. That's the do reweighted 1D.sh. This is the script. So in this script, it's actually calculate the 1D PMF. But in order to use the scripts for remote users, you can just do just run the normal sh script, run 1D PMF.sh, and you get all the results.
What's the output file from this?
Yeah, so now we've generated the 1D PMF. If you cd into this folder, that's a PMF psi, or we use some cutoff 600. So if you cd into this, which I put in the tutorial, so yeah, generates this file. So you just cd into, okay, I think this is for 2D. Yeah, so we just cd into this PMF. Right, so, and then then you see this PMF C2 is the one that we use Gaussian approximation to the second cumulative order. So that's actually the one that follow. That's the one that we usually get some different results like C1, but the most accurate one is the C2. So if you use XN grace to plus the C2, then you should get some, you get these results. The PMF is actually, it looks like this for psi. So for the psi angles and the PMF, but we don't have XM grace in this, like you can't use that here. So but if you copy the files and then you plot it, you should get something like this. Okay. So you can see that the PMF is most energetically favorable at 0. And here you can see different conformation of alanine dipeptide that we're able to sample using just within 90 nanoseconds. So we're able to sample different space of different conformation of alanine dipeptide. So you can see different low energy wells, and also there's another energy well at about 150, which you can see here, like, though it's not really as low as this one. I feel like this is very low, but you can see this low energy before it wants to go up. So I feel like those are also corresponding to this energy state. But if you look at it at about minus 60 to minus 180, you can't really see, like, they are not energetically favorable, and that's why we don't have any low energy states in this region. Yeah, after we have plot for remote users, there's a script, also .sh script is a bash script. Feel free to just run 1D PMF.sh and put hand up, like it's going to run fine, and then you're going to get the same results we are getting here if you use UNC remote accounts.
May I ask a question? I know you routinely use potential mean force or free energies in these plots. Is there some reason it's more natural to think about those things in terms of probabilities? Is there a reason that you don't tend not to do that for plot free energy instead?
I didn't get the question. Like,
well, I tend to think about distributions of conformations in terms of probability of finding. And so, I could think about that in terms of using something like a Boltzmann factor to convert energies into probability. Is there a reason you don't go in that direction and you just plot?
Right. PMFs. So, for the I think like also we consider probability. I think there is a but like during the weighting, we also have the KB from the formula. We use delta G equals to like if I show the scripts. So we do incorporate the Boltzmann factor and also so if I open like do reweight. Yeah, that's right. I'm trying to want the test. So yeah, based on this formula, we actually consider it in terms of probability as well. And then we
Yeah, I was just wondering if there's something wrong with thinking about it in terms of the probabilities. It just, are there some issues that I'm forgetting or unaware of?
yeah, so we actually run different. Number one, we run different replicas to ensure that we get enough statistics in terms of probability that the region, the conformation we are actually sampling is actually real and it's not like something that is not stable. So that's why we run different replicas. And I also think like for the Boltzmann factor, I'm not sure like we don't really, but I think
So yeah, that's in the approximation.
Right.
We have the Boltzmann weighting factor, right?
Right.
Like I mean, we can follow the slides I shared in the morning.
Okay. Right.
Can I add a small comment? Well, the PMF is the logarithm of probability.
Yeah, right.
That's a good question, actually. Why don't we just plot the probability and see what is the most probable conformation?
Yeah.
The only advantage that I can see is that when you plot the logarithm of the probability, you can zoom.
Different.
Yeah, and you can zoom in and compare smaller numbers.
Yeah.
So if you look at the regions where you have like small probabilities, you can't really see it in the probability plot. But if you do the logarithm, it's like zooming in, and you can see that you can compare smaller numbers better. That's the only advantage that I
So it's just a question of visualization.
Yeah, you can do
results. There's nothing very fundamental.
Yeah, different ways to visualize.
You can do.
That's fine. I just wanted to make sure I wasn't missing something.
No, you're right. You're right. You can do either.
So I think we can now run the 2D PMF again. We're going to do SPatch and just run like run 2D PMF.pbs. So we're going to submit this.
So the probabilities are exponential.
So I think it's running. Yeah, so So if we seed into this file, see the PMF, again, we're going to get different results. And then the real results we want to, our focus on this case would be like PMF to the C2, that's cumulant expansion to the second order. Like that's what we want to actually, that's a file. We want to copy the most accurate file. Then we copy that, and then we can use Origin Pro to actually get the 2D PMF as well. So I plotted that, and again, you can now see, and the 1D and the 2D are very like, from the 1D energy profile and the 2D energy profile, you can see like there, you can compare the two in terms of like, if you look at the angles, right? Like for example, 0, we can see that's a very low energy state, and in this case, like, you look at that 0, it's sample different configuration in the for psi. But in this case, this is psi and phi, but I'm just comparing this alone. For the 2D PMF, then we can see different if you get out this phi, this PMF2DC2 phi psi weight, and you plot it, I plotted this 2D using Origin Pro. So you get something like this. So you can see different low energy states for alanine dicryptophan.
So what's the benefit of using or being able to accumulate your PMS according to the first, second, and third cumulants in the expansion? I mean, I think I understand why we're focusing on the second one, because that captures essentially our Gaussian behavior, but like what can we learn from the first and the third cumulants?
Right. So for the first and the from the GaMD paper, they found that the weighting is not that accurate if you use the first order or the third order. But like if you do the approximation to second order, you get a very good result that is comparable to conventional MD. Like,
so if I may,
yeah.
For the first order, it's the same as Maclaurin series expansion. So there's a systematic activation because you are missing the second order. For the third order, it's more noisy because they have very high fluctuations with a high boost.
I thought, maybe misunderstanding too, but I thought the main reason for going to this cumulative approximation was that you're trying to average over these individual frames, right?
Right.
And if you try to work directly with these exponentials energy, they blow up.
Yes.
It's impossible, computationally impossible problem. Right. And so the cumulant analysis, I thought, was really a way of bringing this back into something that is easily computed.
Exactly. Victor, if you can actually go back to your equation.
Okay.
For the exponential average factor, yeah. So over there, yeah. Exactly as what Jim mentioned, like if you calculate the exponential beta delta V, if your delta V gets to like a 50 or 60, that's a huge number. Then your number will become infinity. So you cannot go down
Yeah,
the
you can't represent those numbers in your computer.
Right, exactly, yeah. But with the cumulant, I can resolve that problem.
So if the boost potential is greater than 50 or 60, then we exclude those from the.
It depends.
Okay.
But normally we don't get into that situation. But say for PPI-GaMD, the boost could be pretty high. But still, like if you have enough frames, it depends on the cutoff you use. Yeah.
So now we're going to run the 1D harmonicity again. The script is. So for the 1D harmonicity, there is a script here that is called the run 1D IAM. So we're going to submit that script as well to get to see if our simulation actually follow Gaussian distribution. So 1D. PPS. So we have actually generated the 1D. I use this command. So I think you see a lot of files. So here is the histogram of the psi angles, and here is the harmonicity. And you see some other phi dv stats, but to actually look at to plot the harmonicity, you just need to copy the dv a ham dash psi.x. So if you plot that on XMGrace, you should see something like this. It doesn't look like but again, it's a representative, right? But But another thing like we can pay more attention to is like the value is actually very, very smart, about 0.02. So it actually indicates that because the lower the harmonicity value, the higher the chances that the simulation actually follow the Gaussian distribution. So in this case, I think like you see like most of the energy states actually at 0.002 around it. So the value is actually very low. Yeah. So it shows like how our simulation actually converges and it follows the Gaussian distribution. So the next thing is this is for the 1D. Actually, we can also plot for the 2D. That's the psi and the phi angles. Again, the script is there. We can do the same thing and submit the job for the 2D using the S-batch S-batch 2D, S-batch, so we can do S-batch. So here for 2D, basically we are considering two reaction coordinates, the psi and the phi angles. So yeah, I think it has finished. And then like you can see like we generated another we generated the 2D for the harmonicity using two reaction coordinates, the phi and the psi angles. So in this case, you can see for the 2D harmonicity, all of it is just like very blue, but because it has a very low harmonicity, which of course is very good. And then we can see like if we compare the two distribution together, like they look alike, right? For the 2D PMF and also for the 2D harmonicity. And see like they kind of have the same kind of free energy profile. And that brings us to the end of that. Okay, yeah. Then for the basically for the if you want to look at the boost potential energy distribution, also if you it does irrespective of the reaction coordinate we are considering you should follow a very perfect Gaussian distribution. So as in this case, you see we get for that of for 1D and for 2D, and they are exactly the same. So and this brings us to the end of the tutorial for GaMD. If you have any questions, feel free to let me know. And then like if you have any question, we have a mailing list. Like maybe you run into some trouble while you are running your simulation. No, you can just let us know through this email list. No, we'll be happy to reply and then to help in whatever we can. Thank you very much for your time and for the attention.
We still have time, or we can take a break.
Question, when you're doing those, some of those averages, you're taking segments out of the trajectory, a certain number of frames, or?
No, we are looking at the log phi, the GaMD log phi.
Right. And but you're not averaging over the whole thing. You're breaking it into segments.
Yeah, if you go to reaction coordinates, if you look at the any PMF, right? So you have reaction coordinates. They have a bins.
Yeah.
So maybe every
So
6
what
degrees.
What are the bin
Yeah.
sizes? That's what I.
So you may look at the distance. So if the values fall into that bin, so you have those frames due to the average and the standard deviation. I get the cumulants.
So in this case, we actually use bin size of 6. Yeah, so that's the bin size that we use for both for the psi and also for the phi.
Okay, so they're binned in segments of phi and psi.
Right.
Okay. Yeah.
How do you know when you've run long enough?
Like when we run long enough, production or conventional MD?
Production, like in GaMD.
Okay, production.
Like how do you know your PMF is converged?
Oh, so I would imagine like you have some reaction coordinates, what you really want to see, right? So if you plot that and then you see like maybe for a very long time, you just stay there, I feel like you can just stop the simulation. And
How much time is a very long time?
so I don't think I can put a like very specific to say like this is a time frame, but I think like if you just do the PMF and then you see a very low, well-defined, low energy state, then I think that it's very feasible. Like in this case, I think that should be fine. Everybody is like they can naturally assess that. Oh, it's converged when you plot the PMF.
But that can depend on the system, right? I would say, hey, you get to PMF directly, but if you plot the psi, the dihedral basis time, if you see the transition multiple times, like 3 or 5 times.
So there's like a general rule of thumb for transitions that you observe.
Yeah, I will try to get maybe 5 times. It might be good enough. So Jim also showed like that glycans can have transitions, right? Across different states. If you see the whole transitions 3 or 5 times, it might be good. Less than that, like less information is not long enough. Yeah.
Any other question?
Just related to that, I know a number of people, some in the literature were simply doing averages over successive frames and trajectories rather than binning on a parameter. When they were doing that, the number that I remember is about 500 frames. Does that make any sense? You know what I'm talking about?
Can you say the number of frames again? Yeah.
Well, I recall people trying to, get conformational representatives out of the MD trajectory. And rather than binning based on angles or anything like that, they would simply take the 0 to 500, 500 to 1,000, 1,000 to 500. So your bins were segments of the trajectory. And that, so I mean, that's not what we do, but I know there are some in the literature like that. And I sort of remember quoting sort of a magic 500 frames as a reasonable segment to carry out the average.
So yeah, that's related to clustering. So you have a trajectory, so you have 1 million frames.
Yes.
You can do structural clustering and you have maybe 100 clusters. Then for each cluster, we can also apply the weighting. But to a certain extent, if the number of frames is too small, then it's too noisy.
Yes.
So your accumulants would give you a big trouble.
Yes.
In that case, like we have a cutoff. So the cutoff, it can be 500. If that cutoff is below 500, we say, whatever you calculated, the average standard deviation, those numbers are not accurate. So 500 may be about that number.
So some cutoff in the number.
Yes.
Samples.
Yes.
I think the cutoff is 300.
Yeah, they can change depending on the system you have, right? So you have a very small system. The cutoff can be very small. If 50 frames would be good enough. If like the system is large, like some memory protein, you need to have a larger number of frames for that cutoff.
Yeah, I think that's what the question was about guidelines for how extensive the sampling needed to be.
Yeah, we have sentences in the paper, but sometimes you may need to dig into that part. It may be hard to find the sentences, right? Yeah.
Yeah, no, I think it also depends somewhat on how much sort of pre-processing of your trajectory you've done. For example, we cluster ours into structurally related segments. That I think automatically means you have less noise.
Yes.
Right. So you can maybe have more clusters, fewer members.
Yeah.
If you do some pre-processing. And this, collecting by phi and psi is another way of pre-processing.
Yes.
Where you've now put these observations into bins that have less scattering.
Yes, exactly. Yeah.
Okay.
Yeah, in those bins, in those clusters, the frames are already similar to each other. So you don't have a huge noise for the boost potentials. Yeah.
Okay.
Yeah, we can take a 5-minute break. Yeah, we can continue. The next one. Yeah.
Oh, you do? Okay.
And the license for students may be like
Oh, okay.
60 or 70 bucks.
Oh,
a
I
year.
see. Yeah, we think like I tried to.
Jian, you can get started there.
Okay, I'm glad to share some tutorial about the Ligand 3. As Dr. Miao mentioned, this is a new method that published. In compare with the previous version, we have 3 triple boosts in this method. We have boosted the ligand dissociation and the rebinding and the conformational changes. So there are some additional parameters that we need to pay attention. So for the most command that should be very similar, we will go through here. As we prepared the tutorial, we make them mostly similar. So in the Ligand 3, as we provided the tar file, if you can download from the PDF, then they will give you 3, actually two folders. One it is the simulation, another is the analysis. The similar format as the GaMD. So here I just download one in the UNC cloud, UNC cloud. So we can first to extract it. This file. Also, you can. I put 1 copies in the shared data file folder. If you go to the PDF, you can find here. You can just copy this command to copy the files in your current one. So here, as I said, the framework is very similar. We have simulation and the analysis. First, we go to the simulation folder. We also prepared the batch file, the PBS file to submit the job in the UNC, or the SH file to submit the job in your local workstations. So first, as this Ligand-GaMD, or we started from already a short convention. So we already prepared a topological file and the restart file from the convention. Here, this file, this one, it is the topological file. And this restart file, it is already go to 20 nanosecond convention simulations. If we go, so for submit the jobs for the equilibration, as shown here, it is just using the batch run equilibration PBS file. In your local, you can using your SH file. If we go to details about the file, the command you can see here, it is first the I, it is the input file md.in. This is the parameters that controls all the parameters for both the MD and also GaMD part. And this P, this parameter is the topological file. C, it is the restart file. O, it is like the output file. That's primarily record the energy information of each step. And this X, it is the most important part, is a trajectory file. And the R, it is the restart file that will record it, the latest coordinate and the velocities. That's very helpful because the computer might be not some crash or some time limitations. So you need this restart file to continue with your current simulations. And for this list up, they are the same for all the Amber simulations. The one additional one is for the GaMD, it is the GaMD list parameters. This specific denotes what the log file you want to put. If you're using the default, they will generate the GMD.log file. But for every simulations, you will have the same names. You don't want to overwrite them. So be careful about that. Then you see it's right now is on running. If mostly first we want to check if running correctly. First we will check the MD output file. So in the beginning, if you go here, it generally will record what the input file you are doing. And for the GMD part, first need to check it is here. So there was one information of the GaMD. It will tell you which the input, which the like the IEP, IGMD, stash different modes or the scales you want to use. Because for the initial, as I said, this is the equilibration. So we don't have the booster potentials to read. So that you can see that all they are 0. Here, they are all 0. But if you go to the production, that means you already have known the potentials. This should be some values from the GaMD result.dat files. So as this running will take some time, I just to go through some special parameters for the Lincoln GaMD 3. First thing that I'd like to mention that it is here. In a selective GaMD, no matter the Lincoln GaMD, peptide GaMD, or PPI-GaMD, make sure using the NTF equal 1. This one. Because in the boost, we specifically calculated all the interactions. So this first thing. The second one, it is the black one. This is the special parameters for the GaMD. So for all other things are very similar, like the IGMD controls which algorithm you want to use. For the Lincoln GaMD 3, we're using the 28. And for the IE or IREST GMD, these are the same. According to here, the NTEP pre and the NTCMD pre, this one are the same as the normal GaMD. Additional part is shown here. We have, as I said, we have tribal. So we have another sigma OP, OB, another IEB to control.
Just quick question. Which version of Amber supports LiGaMD 3?
Amber 22 actually is working, but I suggest using the Amber 24.
24, okay.
Yeah. And as in the ligand GaMD 3, we need to define the 2 regions. One region is the ligand part. Another one, it is the receptor part. So the ligand part, it is defined by this TI mask 1 and the SC mask 1. And for the receptor part, we have the BGPRO 2 atom and the NPRO 2 atom. These 2 parameters to define the first and the end of the receptor. And for all other things, others, I think the parameters are quite similar. For here, as I said here, you see the sigma OP. It is very small. It is not like the regional, like the 6, because these methods will highly efficient for the dissociation. If you state these values is very large, the ligands will dissociate, never come back. So this is why I said here it is a small one. Yeah, for the detailed explanations for each parameters, you can find this tutorial. Now, I think we still need some time to. For me, it's already finished. So when you are doing the finish it, we will. For me, I'll check 2 files. First, it is the GMD result DAT file. So make sure, because for the LinkedIn 3, we have 3 triples. So that means the 3 energy terms will be saved in this file. So we have 3 lines. But for the GaMD, we actually have only 2 lines should be here. Another one we'll double check is the output file. So in the end of the file, you will see some information, especially over here. See, it will record which values they are put into the GMD result.dat file. Make sure they are consistent each other for these 2 files. Only difference is the rigid here it is smaller than that saved in the restart file. Has anyone has issues for this equation? If not, I'll submit the productions. So it's the same. So we have another one is the product PBS files. And here I'd like to see what the differences of the input file. So as I said, of course, this one it is also similar as the normal convention MD. So I rest equal 0, NTS equal 1, suggesting that in the beginning, you just generate the atomic velocities by determining the temperature. So that means if you have a different replica, that will generate the different initial velocities, make their more statist sampling. The difference is here. So in the equilibration, we set I rest the GMD equal 0. But if we are going to the production, we should set this one as 1. Then it will read the potentials from the files GMD result.dat file. And if you see here all other parameters like the NT-CMD, NT-EB, NT-CMD PIEP, and NT-EB PIEP, these are equal 0. So that means we don't do the equilibration because these are the control the equilibration steps. The thing is that we need still to keep N-tab equal the same one because this one actually determines to calculate the potentials. And one in mind is that the sigma OP, sigma OD, sigma OB, these parameters is exactly the same as that in the equilibration. Don't change that. Different from the equilibration production. I think this is the most important part. Then if we go to the MD output file, in the beginning, as I said, when you do the restart or the production, it will set that it will read the potentials from the log file. You can see then it will generate the K0P, how to use them to apply the boost in the simulations. Therefore, this one is already fixed in the production. This is most of the we can have a look at. And different from the GMD, from the equilibration, from the productions, from each steps, we will have this boost potentials. For the GMD equilibration, in the beginning, actually it is the convention MD. So for that part, we don't have values about that. Yeah, we can take a break to wait for this simulation done. Then we can go to the analysis part. For the users, you can check if they are the red box. That means the command running in the UNC cloud. If they are a blue box, like here, they actually prepared for the users using their own workstations. Yeah, after they finish it, we can go to the 1D, 2D, 3D PMF.
Your timescales for running ligand GAMD change depending on like how deeply bound your ligand is into the
Yes.
protein target?
Yeah, it's dependent on the system. So that's why we need to tune the sigma OP values. But you can imagine for some very strong binder, you need to add more boost to push out. But if for some very weak binder, you just need a slight boost to do that.
Okay. How significantly different are the timescales for those two types of situations, like say a shallow binder versus a deep pocket?
You mean the time scale for the ligand binding and the dissociation?
Like typical timescale to do your ligand
Do the
GAMD
simulation?
run. Yeah, because I think you mentioned what microseconds for the slide you had this morning.
Yeah, for the
Or one microsecond.
trypsin deflyzine,
Yeah.
we can do 1 microsecond.
Yeah, for the paper we published, we're using the MDM2 bound with the peptide. We have 2 microsecond simulations. Yeah, but for here, this is a very small system. So we actually 300 nanoseconds is pretty good. I just have one version. Oh no, I get it. So for this host gas is a very small system. So 300 nanoseconds is a capture about eight different dissociation and rebinding. So that's dependent on the system. But for the most protein systems, it will be go to microsecond simulations.
Yeah. Okay. And I'm sorry, I need to read the ligand
Oh,
GAMD3 paper, but have you guys had to modify the Kramer's theory formulation much from when you guys initially
No, not
okay, so it's
much.
pretty much still unchanged.
For the kinetic reweighting, it is still the same, yeah.
Yeah, for that part we didn't. Yeah.
Actually, for your question, it may help to add some time courses.
Yeah,
in the tutorial, yeah.
first version we have, but yeah, I deleted it. Sorry.
If you look at the time courses, you will know how many translations you use, and would that be enough?
Yeah.
Yeah, I think I can open it up. You see here, this is about 10 nanosecond. So
yeah.
So this one is quite efficient because this is a small system.
Yeah.
So if anyone finishes the simulations, we can go to the product, go to the analysis. As in the analysis folder, I actually prepared similar for the 1D, 2D, and 3D. Because 3D rewriting we use the letter to calculate the binding free energy. So I just to show that also. Then we also prepared the PBS and the SH, two type of the squares. One is for the local. The PBS is for the local UNC, and for the SH it is for outside. So we can just, if we submit first the 1D PMF, we can see here the command. As this actually for the rewriting, it is the same as no matter which GMD work. So basically we need one Russian coordinate and one the local file. So these two files need to consist with each other with the same frames. This is very important because if you can imagine you're using the long boost on the Russian coordinate, you will get a very strange PMLs. Yeah, if we run the simulations, then we'll generate it one folder PMF the files. Then if we go to the same folder, this will give you the different orders of PMF C1, C2, C3 corresponding to the first, second, and third. And also we provided one calculation for the non-related, just for comparison. So if once we get these files, we can download it. This is what we get in this file. I just provided 13 nanosecond trajectory to generate this one. If you see here, quite interesting is that you see the unrelated and related, it is not a huge difference. That's because for this system, as I said, I tuned the sigma OP is a very small 0.1. So let's give you a very small boost. So that's not huge differences. But if we go to the LinkedIn papers, we get a quite a large boost. The PMF might have quite large differences. Then we can go to the. I'd like to explain a bit of the So the most control it is on this related this one. So you can control in this one we have, as Dr. mentioned, the cutoff. You see here, the cutoff and the bin size. This one it is these two parameters. It is very important for the reweighting. So for the different system, like a small system, maybe you have the cutoff might be very small, but for the large noisy one, we might expect a larger values for the cutoff. And the bin size is dependent on the relation coordinate. For example, the distance, we most use the 1 angstrons, but for the angle, especially for the dihedral angle, we might use about 6. But for some of the native contact, that will be hundreds, something like that. Then we might use the bin size as much 5 or something like that. That's depend on the systems. So you can tune these values here in this squares.
No.
Yeah.
Are you using? I was just looking through the script too. Are you actually using any Amber-based tools in the reweighting?
Reweighting part we don't.
So there, you really don't have to.
Yeah.
Add Amber to your path.
Yeah. No.
Okay.
Yeah, but as I write in the tutorial, we need to install the new NUMPY and SCI pi installed. There are two libraries need to be installed.
Okay.
So the Amber maybe for CPP trash, like Amber tools.
Yeah. So you can get by with Amber tools instead.
Yeah, they are reaction coordinate values. But you can also use some other tools.
I'm just trying to think of OpenMM based.
Yeah,
I see.
For that part is that because the Amber right now, especially from the Amber 24, the Pythons they already installed all the library we have. So that's why if you modulate that, we can just using that Python.
Yep.
So we don't need to install them ourselves because it's quite noisy to me.
Agreed.
So I think the 1D and the 2D. Yeah, 2D will be generated another PMF like 2D that file is same as the GaMD. I just show you the differences of 3D. So that's basically for calculating the binding free energies. Now if you see here, they will generate further and here. And this also we have C1, C2, C3 and the non-related. So I brought this one in here. So you because I'm using the host gas distance and the RG of the host. So to see that was because it's is very short, so we don't see, we can see a low energy state here, a bit of intermediate state here, but it is not a reliable process. I want to emphasize. So another one, it is the 3D, as in the Ligand-GaMD paper, we get these methods. So this is based on the ligand state on the bound state and the unbound state using the populations to get the binding free energies. So we have this squares, the run rewarding 3D. This is different from others. So the results, it is on the output file. Oh, but it's not ready. Yeah. So in a 3D output file, we have that one. It is the DG. This one, it is the binding free energies. But if you see here, the values here, it is the -25. It does not make sense because, as I showing here, only one unbinding showing. So mostly they are bound. So that's why the binding affinity it is really strong. But I calculated about the 300 nanoseconds. It give me about why it is very close to the experimental wall. So just around the one kPa/mol. So that means the status is very important.
So how many binding events should we do we need in general? Maybe 5 or so?
At least for 3. So
at least 3. Okay.
Yeah. Why is better? Yeah, I think this is if anyone can catch it or here. Also, I have one parameters want to. So, except for different from the 1D and 2D, we have first the 1D is from Russian coordinate, and also the GMD file. We also have the 2 parameters. One it is the bound cutoff for the bound. Another one is the cutoff for the unbound. Because we calculated the population of bound and unbound, so we need to state up these values here. And this one it is mostly what we do is to plot the PMF. So let the intermediate states it should be the cutoff from the bound and unbound. That's using the lateral values. And for this one, as I mentioned here, it is the vector. So it is the 3D vector. If you see here, it is calculated by the vector in the CPP charge. So this is quite specific analysis.
Can I ask you a question? So when a ligand is buried deep into a pocket, and it's kind of challenging because when you remove it, you need to see the water going in.
Yeah, water and also some conformation changes of the pocket.
Yeah, so it takes quite some time to do all this stuff in the simulation. Do you observe them? Like if you visualize the trajectory, do you actually see the ligand coming out and the pocket opening up and the water going in? Do you see all this stuff? And then when the ligand is being boosted to go back, do you see the water going out and then the ligand going in? Do you see this stuff?
We do see like the Ligand-GaMD2 is specific for that because for the binding pocket is closed. You can imagine the ligand want to go out, the binding pocket must open. So we do see the open and close during the ligand binding and the dissociation. But we didn't focus on the water, so I don't.
Yeah, not even that, not even close. Like even if it's like just a hole like this.
Yeah,
just seeing the water going in and out. This takes some time.
I believe the water should go inside and
Yeah,
go
but
outside.
you could actually visualize that. You actually have the trajectory.
What
if
you
don't
seen
take
those
the water around.
Let's say question. Let's say the issues for our simulations. We set that only save the protein and the ligand. We didn't save the water, but we can certainly go back.
Yeah, we have water molecules in the restart files, right? But it's true, like, if you save coordinates for all the atoms, we can track the information.
Yeah.
It's just, it takes tons of space.
Too much information.
Yeah, as I said, I explained in a tutorial. We used this one NTWPRT. Let's give you the options. Only save this number of atoms, the coordinate into the tree. So that will save a lot of space. But we also, as we lost the information for the water. Yeah, but I believe the water should go inside through the.
Yeah, if I may add, say, sometimes if the water dynamics is really exciting, you want to check it out, right?
We can.
Then as you select the dissociation binding, you can restart some summation and save all the atomic coordinates. Then you can look at the water again. So that's another approach.
Yeah, for that one, the Ligand-GaMD tool might be more efficient.
But that's probably one of the more worrisome areas of simulation, is how good are the water protein interactions and water ligand interactions. Right. Right. So you could look at it, but you have to also be aware that it's an area that probably has more uncertainty than just looking at the protein.
Did you guys notice any differences, like with your implementation in Tinker HP, with the quality of your results? We're using Amoeba with that.
So Jay and
Jean-Philippe?
Yeah, he did the coding and we have not done the testing.
Oh, okay.
So we don't I don't know.
Yeah.
How that works for different systems.
Curious.
Well, normally for Ligand, as you come up with the method, you want to do quantitative comparison, right? So the first things you want to check would be delta G canonical off. For the water dynamics, certainly it's very exciting in terms of the mechanism, the pathways, right? But that could be time consuming. But certainly you can go back and look at those. Yeah.
Are you going to talk about how you get binding constants out of the simulations? I mean, you've got kinetic.
Yeah, we have the kinetic of the K off divided by K on, we can get the binding constants. Yeah, the KD. We actually do that.
Yeah, you do. Okay.
So we have two ways to get in the binding free energies. First way it is, as I said, the 3D PMF rewriting. Another one it is just based on the K on and K off values. Because delta G equal minus log K off divided by K.
Is there like one way more accurate than the other? Is that why you do two ways?
Sorry?
Is there like one way more accurate than the other when you're calculating binding free energy?
In sometimes this, I'll say for some systems, but the behavior is different. Because for the Ligand-GaMD study, we do see the systematic estimate of both K on and K off. So that give us a very, it is not ideal K on and K off, but give us a very good results of the binding affinity. That is because both are scaled in the prediction. But let me my thought is maybe the force field issues because both K1 and K off are accelerated.
Gianna, seems we still have time, right?
Yeah.
Maybe you can explain how we get the kinetics. If you use maybe open your pic and talk briefly about that.
My order maybe.
Previously we worried we don't have enough time. So the tutorials are pretty clear to follow, right?
You can see the slide. Basically, is that because you know for the kinetic, we know only the unrelated from the trajectory. That K1 and K2 we actually know. We just calculated that on the time courses. We get that K1 and K2 values. We don't know it is the K1 and K2 the related. So what we do it is using this equation. So the Kramer equations for using, you say if say Kr the related divided by the Kr the unrelated, then we can get the different values of these equations. That's based on this one. So on these equations, what we need it is the delta F, the delta F. So we actually get the two delta F. One it is the related, another is unrelated. And another one it is the omega M and omega B. These two it is the second orders of the curve in the PMF in the low energy states or in the bounded states and the unbounded states and the intermediate states. We can get that values from your PMF to get the second order. And all these different things is this how to say. These values we using the small
molecules equation.
Using that. Also we because that also based on the PMF. So we using also get these vectors. Then we get the accelerate vectors. Then to using the unrelated to get the related K1 and K2. Yeah, this one is quite tedious because to be honest, it's quite tedious.
Because kinetics is complicated, right? You need to worry not just the French profile, but also the friction coefficients. I want to clarify M would be the energy minimum, B would be the barrier. The top of your French profile.
So the delta F, if you want to calculate the K-on, it is corresponds to the energy barrier of the O, and then another one of is O. Yeah, if you see here, you can see the PMF for this results, it is totally different because we had quite a large boost. So, if you get a very large noise PMF, it is rewriting my design issues. That's why in the GaMD methods, we also in the paper we also say that try to use in the smallest of the boost as possible.
I'm checking whether we have more questions on Zoom.
What is clear?
Maybe like a late night in Asia. So lots of people go back to sleep.
No, as the that's one question curious about the storages. how do you deal with the MD trajectory? Because we know once we publish it, do we still save that or not?
You have more questions on Zoom?
Yeah. I think for the size of trajectory, for the rewriting itself, we might need millions of frames. But as the simulations, yes, as it said, we do have two strategies. One is we saved a large trajectory in a very small, like 10 steps. Another people might be save the 100 steps, so that will make the size totally different. But as I will say, it depends on the questions you want to address. Mostly for the status, we will still think about if you want to observe some conformation change or ligand binding or dissociation at least 3 times. Then in your simulations, then will give you pretty good results of the PMF. Do you have any ideas?
Yeah, I mean, that's a problem we've been trying to resolve, right? Like, I would say, I mean, if you look at that, say the real time, if you get to, I don't know, milliseconds or second submissions, you can have a large stride, but your data would be also huge, right? Now, yeah, we have like this accelerated submission. Yeah, we have a large, I mean, a very small stride to save the trajectory, and then you have big files. And that, yeah, that's just the reality, you know, like we need to have so many frames to wait and get the French pair files. But still, I mean, if you compare this with maybe some other, I don't know, popular programs, like say AlphaFold, say how much space you need for AlphaFold. Terabytes.
Terabyte, I believe so. Because for the library, it is already terabyte, I believe.
Yeah, no, I was just setting up OpenFold for us, and it was like 3 terabytes just to save their training data set.
Yeah. So, I mean, on one hand, like we wish to save less frames. On the other hand, like to get enough information. That's the trend we may go. Like it's like big data. We need to find solution for that. Yeah.
So, I just had another question with respect to using like the Pyre weighting scripts. Is there like a minimum number of data points that you need in order to have an acceptable signal to noise ratio with respect to calculation of like your 1D versus 2D versus 3D PMS? So like we kind of already talked about how you need like at least 3, preferably 5, like binding and dissociation events to reliably quantify K on and K off rates. But do you need a minimum number of saved like snapshots from your trajectories, like data points, in order to get good calculations?
Yeah. Let's as I said, for the reaction coordinates, it is you can get the time courses trajectory. So it's better to, no matter which PM 1D, 2D, or 3D, you need to capture at least 3 times of that transitions through that reaction coordinate to get a good good PMF. But sometimes, you know, if the protein just in one state, like the bound states, always the bound states, maybe you can just get one.
But yeah, generally speaking, I would say 500,000 to 1 million frames.
500,000.
Yeah.
Okay.
On that scale.
Okay.
Yeah.
That's
like,
that
that could be the complaint, right?
Yeah.
Yeah.
But that's not per trajectory, is it? Is that total in aggregate? Like, say if you're doing like 3.
The total number of frames you get for a system. Yeah. I think on that scale, normally the reweighting should be good enough. You can get a smooth PMF at least.
Yeah. Okay. Sorry, I just thought of another question too. And I'm like Jim, I'm just gonna keep asking questions because this is my only learning opportunity. What if you have like a poor starting conformation for your ligand bound to your protein target? Like, do you guys do any quality control on that?
Right now we started from the bound state.
You start from the bound state or the unbound state?
The bound state.
What if you have an incorrect pose?
I think we also have some systems from the unbound. It should work, but it will more difficult.
I mean, but if
you can imagine the energy barrier. See here. Because mostly the dissociation energy barrier is much larger than the bound. So you can imagine if you go from the unbound to the bound, you can the potential start learned from the equilibration. Actually, it is only a very small energy barrier. But when it go to the bound, once the system go to the bound states, it is really hard to dissociate. That's why we go to the bound simulation in the beginning.
Right, but if you have an incorrect bound pose, then it's going to be an artifact that you're introducing a much lower energy barrier for it to dissociate.
That's a good question. We didn't test that yet, but to me, my personal opinion, it is not so much, not matter so much, because for the simulation, if we can capture both binding and dissociation for multiple times. So that's actually the refining of the initial binding pose. So in that regard, the initial pose it should be not so important.
I think I understand what you're saying. You're basically making the case it comes out in the wash because you're observing multiple binding and unbinding events.
Yes.
So it's not going to significantly alter your populations if you have a poor initial stock complex.
Yeah, it is not. To me, it is not so important. Certainly, it's important, but it's not so important. But if you from the unbound to bound, it is really hard for some strong binder. It is really hard to dissociation.
Yeah.
Maybe a bad initial configuration would be more of a problem if you were using like a geometrical way of getting the free energy, like pulling it with some geometrical restraints, basically. But shouldn't this method be a bit more protected from all these artifacts because it doesn't use a collective variable, basically?
Yeah.
So it allows the system to find its way by just boosting.
Yeah.
Is that correct, actually? I don't know.
Yeah. I may say, if you have a very bad starting pose, it may affect the potential statistics. So as you get the parameters, those parameters may not be the best. So you may just run a little bit longer summation. In other words, the summation may not be that efficient, right? You can run a little bit longer summation and still get the processes you want. but in like yeah, it's hard to quantify that. I mean, it can do some comparisons, right? Maybe using some crystal pose versus doctor pose.
Oh, yeah, I mean, like, that would be your preferred situation, right? So that you have some sort of structural information.
Right.
About your starting conformation. I'm just trying to think in terms of like, if you were gonna try to run this at scale, like on the thousands to millions of molecules, you don't really have the benefit of visually inspecting every single one of your starting complexes. So, yeah. I'll let you on.
We have to start from the bound state.
Yeah, we start from the bound state.
Like, but what if we just have like unbound, like a ligand and a protein, like maybe, or can we just start from there?
If we, as I said, if we go to the unbound sets, it's probably will search some surface, then will at the binding process. It is hard to dissociation. But if you just want to capture the bound sets, that's actually quite efficient. We do have a testing for the ligand put one outside like 20 angstrom outside to see if it could go to the right binding site. Yeah.
A quick answer, okay, like you don't have to start from the bound.
Okay.
Actually, in the initial GaMD paper, we actually started from unbound.
Okay.
We try to capture the binding process, right?
Just wanted to
But
make sure.
it may just take a longer time.
Yeah. Yeah, we don't know the bounds. Always start
Yeah.
from, like 10 things.
Does that affect the training of your boost potential?
Yeah.
If you start with it.
With the unbound, you need to spend lots of time to see the binding process. And yeah, you need to see the entire process so that they have the information about the entire profile. They can train the potential model basically.
Stop.
Yeah, thank you, Jian. I think we're running quicker than the schedule. So
take a rest.
Yeah, do you want to like take a break like 15 minutes? Then we can start at 3:15. Maybe we can end today before 5. So there's something you prefer?
Good question.
Okay.
When you started from the unbound state, did you see a range of conformations of the ligand, or did it always go back to the same place with the same conformation?
It depends on the system.
Yeah.
If you have a challenging system, yeah, the ligand could go to many places. So the ligand could also get stuck in some stable binding sites. So yeah, normally we run multiple submissions to check that. Ideally, like you still want to say, yeah, ligand bind and the dissociation multiple times in each submission. Then yeah, you have a complete picture. Sounds good?
How much?
Okay.
Clinical life on blood infection.
5 lectures.
Oh, wow, that's very light.
About you?
I'm teaching to this quarter, so this is my heavy teaching quarter. Yeah. But the other quarters, well, thankfully for this year, I don't have any teaching, so it worked out with maternity leave. Yeah, so I don't have to. Yeah, I mean, I didn't have responsibilities in the first place. But it's nice, yeah. I could just really leave.
Yeah.
You can like inhibit bacteria, but
I would say like in pharmacology or school of medicine,
contributes like
you need to get more funding for
Oh,
yourself.
right, that's true.
Like do you have like a 9 month or
Yeah, it's 9 months. Yeah.
all the countries like.
Yeah, I guess that is. Yeah, I guess you have to get more grants.
yeah.
This enzyme, it only occurs in that.
Minutes.
You don't carry it out.
That's why it has a selectivity that the antibody has a selectivity to this. So it's been like now the problem is going, because we don't have like enough new antibody.
I got to also ask my bigger questions to you.
Yeah, that's, yeah. Yeah. Even.
We went on multiple ones to in the tutorials.
Oh, this now using
And the poses, the
these discharges.
this.
Waste discharge.
Hope like this is easy.
Antibiotics. Yeah. What is happening there discharging is like raw feed.
It's probably somebody else's not sometimes muted.
Oh.
There's a house.
So if it's the echo, it's not your fault, somebody else's fault.
Sometimes
they transfer.
So we need to figure out how to do that.
now the tutorials is weird, you know.
Oh, it has now become.
As we get to this finally for
Asian countries. Oh, yeah, there are like plenty of discharge from
just like
pharmaceutical companies.
I thought it was just with like agriculture animals.
Yeah, no, near the like the water, like reservoirs, everything, they all contaminated with this.
I've also heard like people are making counterfeit drugs.
Yeah, maybe we can get started. Yeah. So this is our last tutorial for the last part of the day. So yeah, so yeah, she will talk about the PPI-GaMD.
Hi everyone. So for the last part of the tutorial, I'll give a brief like demo on the PPI-GaMD method. And the PPI-GaMD mostly stands for the protein-protein interaction. And as we go through the like the introduction for this method, so like as Dr. Miao mentioned, GMD is an enhanced sampling. And it works by adding a harmonic boost potential to smoothen the system potential and energy surface. And in that case, you can reduce those energy barriers and like overcome the transitions very easily. And based on GMD, we have a new improved method called as PPI-GaMD, which mostly is used for sampling simulations based on two proteins in the system. So in this method, what it does is it has a selective boost on the interaction potential energy between the two protein partners. And mostly it kind of boosts, like the first boost is applied on the non-bonded interaction energy terms between the two protein partners, which mostly includes your van der Waals and the electrostatic interactions. And for the second boost, we have like a small boost which is applied on the remaining potential energy of the entire system. And this method has been demonstrated on the Barnase interaction with the bar star protein. And you can like download this PDB if you want by just using this PDB ID 1BRS. And in this particular tutorial, we also have focused our simulation study on the Barnase bar star complex. So in this tutorial, you will know how to run the PPI-GaMD simulations, and then how to analyze those simulation trajectories. And then as you like analyze those trajectories, you'll also know how to reweight those PPI-GaMD simulations to calculate the 1D PMF and the 2D PMF. And I also want to like mention that for running these PPI-GaMD simulations, the simulation time for the equilibration and the production is kept very small, like closer to 1 nanosecond of simulation. But in like if we have to plot the 2D PMF analysis or the 1D, you normally like for practical applications, you normally need longer simulation timescales. So as we go to the required files, mostly for running the tutorial, you need to, like for the people on the remote users, they can download this file with the tar extension. And on the UNC cloud, you can just go to this directory and like copy those files from the shared data folder. And then you also need the AMBER24 to run the simulation. And to do the reweighting, you also need the NumPy and the SkyPy installed in your own workstations. So for this
tutorial, you'll need, you'll have two folders, the simulation and then the analysis folder. And if we go into the figure 2, you can see like the simulation folder has around 8 files. So it mostly has the files needed to run the PPI-GaMD equilibration and the production, and also the restart files that you get after running the initial CMD run. And then also the input files that you need for running the equilibration and the production simulations. And then if we look into the analysis folder, it will have all the scripts needed to do the reweighting. And also, like if you look into this, so it will have the distance files to do the 2D or the 1D PMF analysis, and the corresponding log file, which stores the boost potential statistics. And then if you're also like a remote user on Zoom, you can also download the reweighting scripts, which are provided in this link. Like if you directly click on this, you should be able to download that tar file. And again, if you are on the UNC cloud, you can just copy those files from the shared data folder. And the reweighting folder should have like 3 different subdirectories to it for calculating your 1D PMF or your 2D PMF and also the 3D PMF. But for the PPI-GaMD simulations, we'll focus on calculating the 1D and the 2D PMF analysis. So now, as we go into the simulations and like running the simulations on the Barney's bar star complex, first step was to kind of generate the system setup. And like due to like time constraint, we have provided all the users with the conventional MD restart file and the system topology file. So, but mostly like if you have to prepare your own system, like based on your desired system, you can use like the TLEAP in Amber or the charm GUI to kind of prepare your own system using these 2 different techniques. But for our tutorial, we already have performed a 14 nanosecond CMD simulation, and like the users can directly use that topology file and the restart file to run the CMD simulation. So now we'll start with running the CMDs, running the equilibration simulations for the PPI-GaMD simulations. So we can go to the, so first we need to copy the PPI-GaMD directory into our own home directory. So we can do that by using the following command line, by directly copying the folder from the shared data directory. But before doing that, we also have to make sure that we have sourced the correct Amber path. So we can just copy this path and then source the Amber path for the UNC people. And then we can copy the PPI-GaMD folder by just copying the folder from the shared data directory. So hope everyone has like copied the directory into their own home directories. And then we can also enter the simulation folder where we will be running our own simulations. So we can just go and copy this path and then enter the simulation. So as we see, we have all the necessary files to run the PPI-GaMD equilibration and the production. And the same instructions will apply for the people who are on the remote users, like on Zoom. So you can like source the Amber path according to your own workstations. And then you all will be provided with a tar file. So you can unzip the tar file in your own home directory and then go into the simulation folder for the PPI-GaMD directory. Okay. So as we can see in the simulation folder, you will have all the necessary files to run the equilibration and the production. So now we can just take those files and perform our PPI-GaMD equilibration. So again, like I mentioned, for running these simulations, you need to make sure that you have the proper Amber source, Amber sourced in your workstation. And to run the equilibration, you will need the topology file which is given by the complex.parmtalk. And then your restart file from the initial CMD run that we performed for the Barney's bar star complex. And then also you will need your corresponding md.in input file to perform your PPI-GaMD simulations. So if like, I'll show these steps for the UNC people, like how to run the PPI-GaMD equilibration. So first make sure that everyone is in the simulation folder. And then you will have the corresponding script to run the equilibration, which is shown by run equilibration.pbs. So you can just take this script and run the corresponding equilibration run. And then you can check the job status also by using SQ command. Yeah, so you can see that like for me, my job is in the running status, and it should take approximately 7 to 8 minutes for the job to be completed. Yeah, so it's still running for the PPI-GaMD equilibration. And then the same steps people who are on the Zoom need to perform on their own workstation. So they can again go into the simulation folder and then chmod the required equilibration script to make it executable. And then they can run the following command line to run the PPI-GaMD equilibration on their own workstations. And so we can check if the simulation is still running. So it's still running from my end, and yeah, so it's closer to 10 minutes remaining. So in that meantime, if someone has any questions about running the simulation, yeah, they can ask me. And also, so the output that you should get is mostly your, like you can also see you should get the md1.out, which basically stores the system energy. So it's an output file that stores the system energies. Then you'll have the corresponding trajectory file for the equilibration run, which is given by md1.mc. And then you also have the restart file given by the gmd1.rst. So mostly you can take this file and restart your equilibration into a production simulation. And then you have the gmd restart.dat, which mostly stores the boost potential statistics and is also needed to restart the equilibration into the production. And with the MD info file, you can see like how much is the time remaining, and you can like track the progress of the simulation run. So yeah, so as this as the process is still going, I'll talk about the MD input file, which is the input file needed for the PPI-GaMD equilibration. So if we look into like these initial parameters, these are all the same as we have a normal conventional MD or a GMD input file. But the major change that we see in the input file for the PPI-GaMD is in terms of the TI mask 1, and then also your BG Pro 2 atom and ED Pro 2 atom. So TI mask 1 and SC mask 1 basically refer to the boost that you apply on the first protein. And then to include the second protein, you put those starting atom, like for this protein 2, the starting atom is 1, and then the end atom for the protein 2 molecule. So those are defined in your TI mask 1 and SC mask 1, and also the BG Pro 2 atom and the ED Pro 2 atom. So these are like the most important parameter as we go from GMD to your ligand GMD or the PPI-GaMD. And then also like we have to make sure like the IGMD mode is 17, which is the dual boosted mode for the PPI-GaMD simulations. And then, so for the testing, you can also keep like the default parameters set for sigma OP and OD around 6. And also you can keep the default settings for IEP and IED, which is 1 and 1. But for my testing, we have switched the IEP to upper bound condition like 2. And also like these parameters we got after doing testing. So like this parameter is like the optimized parameter where you should see the dissociation and the rebinding if you run it for a longer simulation. And I'll just check how much time is remaining. Yeah, so it's still closer to 8 minutes. So in the time as it runs, I'll also brief about those input parameters that we see in the md.in file. So as I mentioned, the IGMD mode, if you put it as 17, it's a dual boosted PPI-GaMD method where you apply the first boost on the 2 protein-protein interactions. And then you also have a second boost on the remaining potential energy of the entire system. And then you also have the IEP and IED. So as I mentioned, like you can have the default settings with IEP and IED as 1. But for my system, I have put it on, I have set it for 2 to have it on the upper bound conditions. And then for IRIS GMD, so this parameter is also important. If you have IRIS GMD 0, this means it's a new simulation. And you will see that as we extend the equilibration to the production, the IRIS GMD parameter is set to 1, which basically means that you're restarting the equilibration into the production simulation. And then you also have like steps where you like calculate how many steps you need for the initial conventional MD and also the initial equilibration. And then you also need to optimize like the sigma OP and sigma OD parameter for your own system. So like you can see like here I have sigma OP 2.7 and sigma OD 7.0. So this is like an optimized parameter set for the Barnes bar star system. But like all the users, like if they are working on a new system, need to optimize these 2 parameter sets. And then, like I mentioned, for PPI-GaMD, the most important parameters will be defined by the TI mask 1 and the SC mask 1, which mostly includes the boost on the first protein on the first protein molecule. And the second protein, like you specify it by using the BG-Pro2 atom and the ET-Pro2 atom. So you can just mention the starting and the end atomic number for your protein 2 molecule. And yeah, and then I think as Victor discussed for the GMD tutorial, like how to calculate these parameters like your NTAV or like how to get the number of steps for the initial CMD or also your equilibration. So mostly for NTAV, we calculate it as 4 into the number of atoms that you have in the system. And then you can calculate using the NTAV parameter, you can calculate the number of steps you need for the conventional MD or your equilibration run for the PPI-GaMD simulation. And it is also important to mention that for PPI-GaMD simulation, you need to keep the NTF as 1 and barostat as 1 to kind of control the pressure settings. So I'll just check how much. So it's still closer to 4 minutes remaining. So is everyone able to run the equilibration on their systems? Okay.
Mind repeating the mask selection flags?
Yeah.
They are a little bit confusing, like just a tiny bit.
Yeah, maybe. Yeah, so if you see like this.
Yeah, these guys.
File, yeah, so the TI mask 1 and the SC mask 1. So like these are the like residues for the protein 1, which in my case, I have put the bar star residues in the TI mask 1 and SC mask 1.
So this is the entire bar star protein.
Yeah. And then for the second protein, like you specify it in terms of the number of protein atoms. So you can specify like the starting atom for the protein 2, which is the barnase. So it starts from 1 and then the end atom for the barnase protein, which is 1727. So mostly, yeah, like these two parameters will, like you need to specify your protein 1 and protein 2, where you want to apply that boost.
Talking about optimizing your
Yeah.
boost potential levels for your PPI complex. Like, is that, I mean, I'm sure it's system dependent.
Yeah.
But I guess, are there any other ways to try to estimate like what general range of boost potentials you may need? Like, say, for example, shared surface area between the PPI or?
Like, so are you talking in terms of like sigma OP, sigma OD parameter or?
Yes.
So normally, yeah, I mean, like for the initial testing, we kind of tested from the default parameter like sigma OP6 and sigma OD6. And also with the IEP and IED as one. But then it also depends, like I think tuning this parameter depends upon the problem that we want to solve. So like, I think as Dr. Jinan mentioned, if you want to see the dissociation, so we kind of increase the sigma OP with also IEP with the upper bound condition S2. And if you want to see, let's say the binding, then you don't need that much of a high sigma OP parameter.
Okay. You guys reached like any upper limit? That just seems, I mean, like, are you going up to like 50 kcals per mole? Or is that just completely unrealistic if you're going to try to model dissociation?
So do you mean like if the IEP is with the is set to 2, then is it like reaching 50 kcals per mole?
Or even like your sigma OD? Like, do you ever have to set that high? I mean, like, what's a?
So I think for like to see the dissociation and rebinding for this system, I think this was like the optimized set which we got. And that's why we kind of extended this one into the production.
And then have you guys tested any, like, bar-star barnase is kind of like the benchmark system for modeling PPIs. Have you tested against anything like, say, alpha-fold multimers or any other type of protein-protein complexes, maybe like some PROTACs or something like that?
I think right now, like I'm kind of doing some testing with like on a big protein, but it's a peptide complex. So, I mean, the peptide is around 23 or 24 residues long. But yeah, I don't think so. We have tested it like this method on a protein-protein complex. But yeah.
Yeah, bar-star barnase would be the most studied.
Yeah.
For other complexes, I mean, we checked some, say, GPCR, G-protein dissociation. And then actually, if I may go back to your question, like, if you set the IEP, IED equal to 1, then when we look at the sigma OP, sigma OD, when you get to a limit, like, say the effective harmonic force constants, like P, KOP, KOD, they will reach 1. Beyond that, it does not help, no matter how much you increase like a sigma OP or sigma OD. Then the difference goes to, say, if you set IEP equal to 2, you can really have like a high acceleration on the protein-protein interaction, right? I think, yeah, normally like we'll do some testing in the equilibration simulation. You can tell like how soon the proteins could dissociate. And with that, I mean, what I'm saying, ideally, if you can say both the dissociation rebinding in the equilibration, then you will know that the parameter set is ideal.
Yeah.
Otherwise, like you want to do more testing, like that could be, it can be tricky as you get a new system sometimes. But I think the parameters we need to tune are not that many. So once you do a couple of testing, you can probably find the best parameter set you may have.
Sure. But you're still gonna need to test it on a system by system basis.
I mean, right now I would say this is still in the beginning of the method, right? As we apply the method on more problems, we may figure out more patterns. So then, that'd be easier to go. Well, now I would say for protein like dissociation rebinding, at least we need to have IEP equal to 2, the upper bound. Then you test more parameters with sigma OP. So that may be the good starting point. So they can narrow the range, Yeah.
Good.
Okay. So I think I've completed my equilibration. So everyone should have these files generated, like the restart file, and then the .dat file, and also the .out and the log files. So now we'll try to extend the equilibration, like the PPI-GaMD equilibration, into the production. And again, for running the PPI-GaMD production, the difference is with respect to the input file. So you need a new input file, which is called as GMD restart.n. And then you can take the restart file from the equilibration to basically extend that equilibration into a production. So people on the UNC cloud can simply use this command line called as sbatch run production.pbs. And then we can check the time. So for me, the job is in the running status. And it might take, I think, another 8 to 10 minutes to get completed. And then, then for the people on the Zoom can also run the production in a similar way, but they can use a different script called as, they can make sure, they'll make sure that they are in the simulation directory. And then they can use this script run production.sh to submit the production job. So as the PPI-GaMD production is running, I'll talk about this input file to extend the equilibration into the production, which is called as the GMD restart.m. So again, the input file looks similar to a GMD file or a normal conventional MD file in terms of all the other parameters. But the only difference will be in terms of the parameters that I've highlighted in lab. So again, like I mentioned, that you need to specify the protein 1 residues in the TI mask 1 and SC mask 1. And for the protein 2, you can specify the atoms, like the starting atom and the end atom for the protein 2, which is Barnase in this case. And then for the production, the iris GMD is set to 1. So as you can see, if you have the equilibration file or equilibration input file, the iris GMD is set to 0. So this indicates that it's a new simulation. But as you extend or as you continue the equilibration into the production, you can just set the iris GMD to 1, which indicates that it's a restart of the equilibration file. And then again, we have to make sure in the PPI-GaMD production, so we are not calculating any statistics for the conventional MD or the equilibration. So all these parameters are set to 0. So you don't collect any statistics for the equilibration or the conventional MD part. And then again, like the other parameters like sigma OP, sigma OD, or your IEP, ED are like similar to your equilibration input file. And okay, so it's still showing around 8 minutes. So is everyone like in the room able to submit the job for the production? Okay, maybe we can like skip to the analysis part because we won't be using like the trajectory files generated for the production from this to do the analysis because it's like around like the production simulation time is just for 1 nanosecond. So it won't be like enough statistic to do 1D or 2D PMF analysis. So we can just go into the analysis, like simulation analysis. So for your simulation analysis, we generated a trajectory file for about 50 nanoseconds for the Barney's bar star system. And you will need like the ones that I've highlighted in black. So you will need like these 3 files to do the simulation analysis. Mostly like the distance between the R59 and D39 residue. And then the distance file between the K27 and D39 residue. And then also the corresponding log file to perform the reweighting for 1D and 2D calculations. And as mentioned here, we also have one script which like I can show it here. So we also have this script which the users can use if they want to generate like these reaction coordinate files. So mostly like for the like due to time constraint, we won't be generating these files ourselves, but you can like directly use those files. So, but if in future, like the users want to generate their own reaction coordinate file, they can just take this analysis.in script and then run it as a normal CPP-Traj script. So now we'll try to focus our analysis on calculating the 1D free energy profile. So to calculate the 1D free energy profile, you will need just one reaction coordinate, which we have specified in terms of the distance between the R39 residue in Barney's and D39 residue in the bar star protein. And then you also need the corresponding log file and then the required reweighting scripts to do the 1D PMF analysis. So make sure like all the users in UNC and on Zoom are in the analysis folder. So now we'll focus on running the 1D analysis for people on, for like people in UNC. So as I said, like make sure you are in the analysis folder and you have all these 15 files. And make sure like you have mostly like the 3 files, like this distance file and like the second distance file and then the log file. So we can just use this command to perform the 1D PMF analysis. So, okay, so my job is still running. And then the people who are on Zoom can also do the 1D PMF analysis in a similar way. So like make sure you're in the analysis folder and then you can just use this run 1D PMF.sh script to run your 1D PMF calculations. So let's see. So as you run like the 1D PMF analysis, you will be able to generate this folder called as PMF with that particular reaction coordinate. And for this protein, I have considered like a cutoff of 50 and bin size of 1.0. So as you enter this folder, you should see these 3 files. Yeah, so as you enter this folder, you should see these 3 files. So you have one file which corresponds to the unreweighted PMF, and these 3 files which corresponds to the reweighted PMF. But mostly for our plotting, we use the PMF, like the reweighted PMF, which refers to the cumulant expansion to the second order. So like the users can like take this file and like can download this file and use Origin Pro to plot this file. So as you plot this file, you should get a figure like this, which is like mostly for the Barney's bar star distance. And you can see the 2 lines for the reweighted and the unreweighted 1 dPm. As we complete like 1D PMF analysis, we'll go to the 2D PMF analysis. So for 2D, like as the name suggests, you will need 2 reaction coordinate files. So you will need the R59 to D39 distance files. And then the second distance file is for with respect to K27 and D39 distance. And then also you need the GMD all.log and the scripts to perform the 2D PMF analysis. So again, like make sure you are in the analysis folder, and people in the UNC can just use this command line called as sbatch to calculate the 2D PMF profile. So as the job is still running, people who are on the Zoom can also perform the 2D PMF analysis in a similar way. So you like make sure you are in the analysis folder, and then you can just use this command line and the following script to do the 2D PMF analysis. So as the 2D PMF analysis gets completed, it will generate this folder called as PMF2D. And then for this also, we have considered the cutoff as 50 and bin size as 1 for X and bin size as 1 for Y. So as we enter this folder, again, we will have, like 1 PMF for the unreweighted, and then 3 PMFs for the reweighted calculations. And mostly we consider the PMF with the cumulant expansion to the second order. So the users can like just take this file, which refers to like the C2, and then use this to plot the free energy profile. And like I've taken this file and plotted the free energy calculations for the distance between the Barney's bar star residue, and again like the distance between the Barney's bar star residue. And like as the simulation is short, like you cannot see like so many energy states as you see, let's say, in this figure. So if you consider like this free energy profile, it's from 2 microseconds of simulation, where you can like clearly see like different states as bound or like 2 intermediate states, and then the unbound states. So again, like if you like run longer simulation timescales around 2 microseconds or 1 microseconds, you can also see like the same states as we see in this free energy. Yeah, I think that's all I have for the PPI-GaMD tutorials. And then if like anyone has any questions that they want to address for this tutorial, like they can just send an email to the GMD mailing list.
That's clear. Any more questions for Kia right now? Yeah, this goes more smoothly than what we thought. Yeah. Well, if not, yeah, we can end it now. And thank you so much, everyone on Zoom. Should I whether they can still hear
I
us?
think like maybe you need to unmute. Maybe I can stop sharing. Or you can come.
Secondly, yeah. All right. Well, thanks so much, everyone. Yeah, yeah, it's the end of the day, and we can finish this maybe 1 hour before the schedule, right? It's kind of earlier. But I think it's a wonderful day, and I hope you like enjoy the talks in the morning. If you have any more questions about the tutorials in the afternoon, I can feel free to email us. Or you can send a message to the GaMD mailing list, so we can try our best to resolve the problems.